Good morning. I'm Jeff Hicks, and I'd like to welcome you to day 3 of the 34th Canadian Student Health Research Forum, and I'm very pleased to say the 5th anniversary of the Gairdner Symposium. Today, we'll go on a journey to discover the intricacies and the orchestration of human development. We will start with how stem cells drive both tissue complexity in development and regulate normal tissue homeostasis in the adult. We'll discover how a simple duplication of a chromatin remodeling enzyme changes brain development and causes a broad range of severe neurological disabilities. We'll then hear about how milk not only provides nutrition, but also provides protection against infections during infancy and lowers the risk of asthma and obesity later in childhood. And finally, we'll hear how the impacts of the pandemic and other social stressors can drive mental health problems in young people and the importance of responding to this challenge. From the Gairdner family, we'll hear research excellence from some of the world's most inspired researchers and a little bit about their journey from where many of you sit now to where they stand before you today. As I mentioned, this is our 5th Gairdner Symposium, and it's with great pleasure that I welcome Janet Rossent, President and Scientific Director of the Gairdner Foundation, and a good colleague of mine, to bring us welcome from the Gairdner Foundation. Janet?
Thank you, Jeff. It really is a great pleasure to be here. Yes, 5 years of partnering with Gairdner and this forum. It's been a perfect partnership as far as I can say. So the Gairdner Foundation, for those of you who don't know, and you really should know because this is a Canadian icon, the Gairdner Foundation has been giving out awards for research, international awards for biomedical research that has impact on human health. And that's been going on since the 1950s when the foundation was founded by Jim Gairdner, who felt that it was important to recognize that those kind of transformational research discoveries that really are changing how human health and human well-being is going to be better for the future. And in those years from the 1950s, we've given out 390 awards. 95 of those have gone on to receive the Nobel Prize. So this is really one of the world's most prestigious awards for health research. And you're going to be hearing from some of the laureates of the Gairdner Foundation today. But when we give out the awards, the mission of the foundation is, of course, to celebrate excellence. But we want to also use that celebration to convene leaders to discuss important issues around research and science going forward today, and of course, also to inspire the next generation. And it's a combination of the last two, convening leaders and inspiring the next generation, that makes this partnership with CSHRF so important to us and so perfect, because you are the next generation. You are the graduate students across the country whose research is now driving the science of the future. And I've had a chance not to see all the posters, but I've certainly looked at the ones that we're going to be judging later this afternoon. And it's amazing the breadth of research that we see in health research across the country in Canada today. CSHRF is our partner here. CSHRF has the 4 pillars of research, and we're seeing research from all 4 pillars being presented at this forum. So really, without further ado, I would like to hand over back to our hosts and encourage you all to enjoy the symposium today. And please join us in the mentoring sessions tomorrow. Thank you.
Thanks, Janet. We have an action-packed day for you today. That means we're going to have to really stick to our schedule. So we will hopefully have time for questions at the end of each lecture and a longer time for a panel discussion at the end of the afternoon. We'll show you in a second the link to the Slido app. Because you're on YouTube, this is the way you can chat with us on Zoom, and you can ask the presenters any question you want at any time during the day, and your question will show up to everyone on the Slido app. If you like a question you see, you can hit the like button, and this will help the moderators to realize which questions are appealing to more of you than not, and they may choose to ask that question in priority. Okay, so let's go. I welcome Dr. Suhaila Karimi, Professor of Physiology and Pathophysiology and Principal Investigator of the Spinal Cord Research Center, to introduce one of our newest Gairdner lecturers. Suhaila?
Thanks, Jeff. Yeah. This is my absolute pleasure to introduce our first esteemed speaker, Professor Elaine Fuchs. Professor Fuchs is a world-renowned scientist for research in the biology and molecular mechanisms of mammalian skin and skin diseases. She received her Ph.D. in biochemistry from Princeton, followed by postdoctoral research at MIT. She then joined the faculty at the University of Chicago in 1980. And in 2002, she relocated to Rockefeller University, where she has been the Rebecca Lansfield Professor of Mammalian Cell Biology and Development and an investigator of the Howard Hughes Medical Institute. Her exceptional career and her transformative research has been recognized by a long list of coveted awards and honors. In addition to being named a Gairdner Laureate in 2020, Professor Fuchs has received the USA National Medal of Science from President Obama, a L'Oreal UNESCO Award for Women in Science, Albany Prize in Medicine, Macchione Award for Innovation in Stem Cell Research, and the Klaus Award for Cancer Research, among many others. Professor Fuchs is an elected member of the National Academy of Science, Royal Society, and National Academy of Medicine, and she is the past president of American Society of Cell Biology and International Society of Stem Cell Research. She has published over 350 articles with nearly 60,000 citations and an impressive H-index of 139. So you can then imagine how accomplished she has been. She has mentored over 100 trainees, and her research investigates how stem cells make and repair tissues and how they evade cancer therapies, causing relapse of most life-threatening cancers. Her today's talk is titled Tissue Stem Cells Survival of the Fetus. And without further ado, please join me to hear from Professor Fuchs about her research. Thank you so much. Please.
Great. Thank you very much. It's so good to see you, if not only virtual. Hopefully that will soon change as the world becomes more and more vaccinated. So it's just a pleasure for me to participate in this Canadian Student Health Research Forum, and it's a pleasure to be able to tell you about some of our work. I'd like to include some of our recent work, but I'd also like to put it in perspective. I always like to start my audience with a few pictures of usually a recent vacation that I took, in this case, back in January 2020, before the pandemic shut us all down. And the Galapagos Islands, I think, serves as a very good example for how nature has clearly had a lot more fun and fancy in creating body surfaces than she has in creating any of the ugly organs that most scientists work on. There's just an endless fascination about our skin. And my laboratory has spent a good deal of my research career working on the skin. I got interested in skin biology back when I was a graduate student, listening to a seminar by then Howard Green, who was then at MIT. And Howard had developed a method where he could take a small piece of human skin and put it into culture, and rapidly was able to expand those cells and transfer them and make sheets of epidermis. I was just amazed. At the time I was working on bacteria, I went to his laboratory to train, and and I've continued to work on skin ever since. I just mentioned that this methodology, really a breakthrough of the realization that stem cells are dependent upon other cells for their sustenance, and using a fibroblast feeder layer was really the breakthrough that then ultimately led as well to the development of embryonic stem cell culture in the early 1980s. And when I started my laboratory, we were already making three-dimensional organoid cultures to grow human skin in the laboratory. My advisor developed the method for the treatment of burn victims with this methodology by taking a small sample of the good skin, expanding it, rapidly developing sheets of epidermis, and then grafting them onto the burned skin. So we really have a 40-year success of the use of purified stem cells in a clinical setting. And one of the fascinating things that we learned about this way back then was that only a few stem cells are necessary for an entire body replacement of skin epidermis, an enormous reservoir of proliferative potential. When I began my laboratory, I was interested in the major structural proteins that these cells make. We didn't know very much about them. We knew that they were called keratins, and my laboratory cloned and characterized the cDNAs and the genes for the epidermal stem cell keratins and the keratins that are produced when they differentially, when they commit to terminal differentiation. We went on to demonstrate that both in humans and in mice, that when keratin, the keratin network is compromised in the cells, that blistering skin diseases arise. In the case of the stem cell defect, all of the stem cells ruptured upon mechanical stress. So just applying a small amount of pressure to the skin surface, in this case for the animal walking around the cage, but in the human child, washing the child's face was sufficient to create this rupturing of the stem cells, and that led to a blister. So we went on to demonstrate that there were other structural proteins associated with the cytoskeleton, and that the cytoskeleton of the skin epithelium is really providing this beautiful infrastructure that gives the tissue at our body surface the integrity that allows it to withstand mechanical pressure. So I just fast forward now some multiple decades to the use of this type of culture technology and now gene editing with regards to treatment of some of these disorders that I had worked on back when I first started my career. At the time, the technology just wasn't sufficient to be able to correct these disorders. Some of them were autosomal dominant disorders. We didn't have gene therapy. And now we can take the skin of these very badly ruptured disease blistering cases. And this just shows you what happens when this child, the same child that you just saw a minute ago, this child now has been treated with the therapeutics after gene editing to correct the mutation causing the blistering. And so this type of approach has really, I think, been very gratifying watching it unfold over all of these years. But more recently, my laboratory has really focused not just on the infrastructure that the epidermis creates, but also on the fact that our skin epithelium is at our primary, it's a primary environmental interface to our body. And our body is subjected to all sorts of different stresses, not only mechanical stress, but also ultraviolet stress, oncogenic stress, commensal and pathogen microbes, wounds, noxious agents, temperatures and pH extremes. And the tissue stem cells have to be poised to be able to sense these changes and rapidly adjust. And so now if we think about a world where our climate is constantly changing, and I'm afraid not changing for the good, Plants are altering the pollens and allergens that they generate. We know that carbon emissions and air pollutants are becoming more complex and more numerous. We know that the ozone layer is thinning. We know that existing pathogens and allergens and microbes are shifting their territories. We also know that there are new pathogens, allergens, microbes, and pollutants that are emerging in our world, as clearly evidenced by the SARS-CoV-2. And bearing the brunt of these environmental changes is the skin epithelium. It's our body's barrier to the external environment. And so we're particularly interested in knowing more about how to protect and how the body does it, but then also what can we do to be able to enhance that protection. And so there's a myriad of immune cells that are permanent residents of homeostatic skin epithelium, and also not only in the epidermis, but also in the dermis. There are a whole variety of different constituents. The ones in the dermis, in the epidermis, reside within the stem cells. The stem cells are in the innermost layer, and we know that dendritic epidermal T cells, we know that Langerhans cells, that they are basically a part of the epidermis, and that there has to be a tremendous communication, because when the skin barrier is breached, stem cells have to be able to commute with resident immune cells. Those immune cells then relay the damage and pathogen information to dermal immune cells, and they then recruit more immune cells from the circulation and nearby lymph nodes. Together, the stem cells and the immune cells have to mount an inflammatory response to be able to first fight the infection, which is a prelude to be able to repairing the damage. The damage cannot be repaired unless the infection is silenced. And so we're interested in how this happens. We identified and characterized the stem cells of the epidermis many years ago now. We know that they reside in this innermost layer of the epidermis. We know that the epidermal stem cells are at the interface between the epidermis and the dermis. They're separated by a basement membrane that the epidermal stem cells make. And we know that as the stem cells periodically commit to terminally differentiate and move outward toward the skin surface, that they undergo a series of different transcriptional changes. But this culminates in the cessation of transcription, the loss of all the nuclei and organelles, and the formation of these dead, flattened, so-called squames that are then continually sloughed from our body surface, continually being replaced by interlayer cells moving outward. And we know that it is these flattened squames that constitute the barrier to our outside world of pathogens, microbes, allergens, and environmental extremes. And so we also know that if the stem cells malfunction and they fail to either make the barrier, or they fail in their communication with the immune system upon a barrier breach, that chronic inflammatory disease occurs. So let me first talk about a very common, the most common inflammatory skin disorder called atopic dermatitis. We know that the treatments for patients that suffer from atopic dermatitis are largely immune suppressive drugs, general drugs that basically harm the tissue as well as affect a whole variety of other tissues as well. So on the other hand, they do suppress the atopic dermatitis. We know that there are environmental contributors, cold and dry environments. And we know there are genetic determinants. Originally, everyone thought that this was going to be strictly an immune disorder. And in fact, people have found mutations associated with some interleukins that are involved in inflammatory responses. But intriguingly, a number of mutations in a protein, a very large protein called filaggrin, a large protein of unknown function that is found in these green granules of the outer skin layers, turn out to have mutations that are associated with AD. those mutations are scattered all along the protein. The protein is comprised of large repeat sequences that are histidine rich. And we know from our recent studies that filaggrin assembles into fluid oil-like granules. So these granules actually are not rocks, but they're fluid-like granules. They become more and more viscous as they grow larger and the filaggrin concentration becomes denser. And then what is really interesting is that suddenly they disassemble. And that disassembly happens at the same time that the nuclei and the organelles are lost and the barrier, skin barrier of flattened squames is achieved. What we learned is, surprisingly, these mutations in human filaggrin that are associated with AD end up abrogating filaggrin's capacity to undergo these liquid phase transitions, as we demonstrated by biochemistry and genetics. And we showed that without filaggrin, nuclei and organelles are retained, and the risk of barrier breach and inflammation escalates as these flattened squames cannot be produced. So really what we're dealing with is then a chronic inflammatory disease that is a disease both rooted in the skin barrier as well as rooted in immune defects. And that turns out to be a paradigm of a whole variety of different chronic inflammatory diseases: psoriasis, atopic dermatitis, chronic wound healing in the skin, also asthma in the lung, inflammatory bowel disease in the intestine. And in all of these cases, as we learn more and more, we're learning that it's really a two-way street involving both the tissue and its stem cells, as well as the immune system. So an interesting aspect about psoriasis and atopic dermatitis, the two most common skin disorders of inflammation, are that the hyperproliferation that is manifested in the epidermis comes and goes, but upon the next assault, it often recurs in the same spots and with increasing severity. You also notice that where the skin is often most affected are in flexural joints, again suggesting that the barrier is a point of importance here, because the barrier is mechanically stimulated and in a negative way at flexural regions. We know that the skin retains memories of its inflammatory experiences. So with each inflammatory assault, the assault comes, it goes. The next time it's more severe. So there's some kind of memory lodged in the skin that is remembering that it's seen inflammation before. The memory endows the skin with the ability to respond faster and more robustly upon subsequent inflammatory assaults. And this can be beneficial in the case of wound repair. In a wound, it's much better if you can repair the wound the next time you get wounded. But it can also be detrimental, which it is in inflammatory disorders. The inflammation can become very severe, and people end up suffering to a much greater extent than the first time they ever experienced the inflammation in these disorders. And so we wanted to study what the underlying basic mechanisms are that account for this amazing memory that the skin has. We first, in the work of Shruti Nayak, who's now an assistant professor at NYU, and Samantha Larson, who was a Ph.D. student in my laboratory, we simply wounded the animal skin and followed it and realized that within about 7 days, wound closure has occurred. And yet if we first exposed the skin to inflammation, and now we let the skin resolve, the inflammation subside, and now we wounded the skin again, we found that every time we did this, the wound always healed faster the next time around. So we then did this by treating with a psoriasis-inducing inflammatory assault first, and again, the wound healed faster after the inflammation subsided. We did this with an atopic inflammatory stimulus for AD. We did this with tumor-promoting agent, a wound for first and second assault, and with yeast as the first assault. And in every case, we realized that the inflammatory assaults can differ, but the response is always the same, a faster inflammatory response when in subsequent encounters. So immediately it comes to mind adaptive immunity, because we've known for years that B and T cells have receptors that can rearrange genetically and become targeted to a particular bacteria or a particular virus, for instance. But in this case, and it turned out from using a RAG2 null mouse that's lacking B and T cells, that the memory is not linked to adaptive immunity. And we could demonstrate that by showing that in the mouse, if we treat with the inflammatory stimulus when the mouse lacks B and T cells and now wounds the skin after inflammation resolves, then we still see a faster wound response. And you could kind of suggest that that wouldn't be the case, because the inflammatory stimulus in adaptive immunity is very specific. It's a particular bacteria recognized the second time around by a particular rearranged B cell receptor. But in this case, there's this flexibility with regards to the response, a broadening of the response. We also eliminated a certain type of T cell and innate immune lymphocyte that is responsible for triggering the psoriatic-like interaction. And we also did the same experiment with macrophages. So we're sticking with the psoriasis-like model in this case. And in all of these cases, we still found that the skin had this memory. And at that point, we began to realize that actually the epidermal stem cells themselves might be possessing a memory. So it's not to say that the immune system doesn't possess memory. It clearly does. But it is to say that the epidermal stem cells still are able to trigger a memory irrespective of the immune system. So interestingly, with few exceptions, gene transcription returns to normal in epidermal stem cells after inflammation resolves. So looks like everything is fine, transcription is fine. But what about chromatin? Does it too return to normal? In this case, we used a technique that for those of you who work in transcriptional control will know. I'll just explain it very briefly. It's a technique called ATAC-seq, which is stands for transposase accessible chromatin with high throughput sequencing. It's a mouthful, but what it means is that if you have chromatin inside the nucleus and it's bound by histones and it's bound by certain transcription factors, that it creates certain accessible regions that are more open. And those can be detected by specific transposase that can mark and tag these regions. And we can fish these regions out then and determine where are the accessible regions of chromatin based on these peak reads, as you will. And so we use this technique to look at inflammation at the peak of inflammation, no inflammation, or after the inflammation has resolved. And what we found is that there are tremendous numbers of changes in chromatin and chromatin accessibility that happen at the peak of inflammation. You might imagine that because there's hyperproliferation, there's all sorts of various different changes going on in the skin. But 30 days later in the mouse, the inflammation is no longer detectable. The pathology looks normal. The transcription looks normal. But we found that even there were peaks that were opened up during the course of inflammation, as judged by this treatment with a drug called imiquimod that generates the psoriasis-like response. And 30 days later, we could still detect about 2,000 peaks in the chromatin. So that's quite a few. And even 6 months later, we could still detect peaks in the chromatin. So here, 6 months after the inflammatory exposure, we're now still detecting changes that are happening at the chromatin level in the stem cells. And as I said, most of the genes associated with, in this case, these so-called memory domains are not transcribed in the interim. Interestingly, if we now wound the animal's skin and look at, is there something special about the genes associated with these peaks? And there is. These genes are rapidly activated upon a secondary assault. So within 6 to 12 hours after wounding, these genes are activated. So there's something special about having these memory domains. We demonstrated that further by cutting out these memory domains and using them as enhancers to drive a fluorescent protein, EGFP. We put our transgene into the amniotic sac of a 9.5-day embryo. The lentivirus that is containing this transgene is then rapidly taken up by the skin epithelium, which is just a single layer of skin epithelial stem cells at this early time, and then we allow the mouse to grow and become an adult mouse. And now if we treat that mouse, if we look just at the control state for these 3 different enhancers, we find no inflammation, no activity. But now if we treat with the stimulus that creates or triggers the inflammation, now we find that these enhancers are sufficient to drive EGFP expression. So effectively, these memory peaks that are harbored within the chromatin long after inflammation has subsided are inflammation sensing memory domains. They are triggering activation of genes long after and able to do so long after the first encounter has subsided. How do we think this works? Now this is the work of 3 graduate students, not only Samantha Larson, but also Chris Cowley and Raj Sejath. And there we have a model, which we're working on and testing, where you've got an inflammation-induced gene, and in the naive state, it's in a closed chromatin condition. And now you have inflammation. So you activate inflammation transcription factors, such as some of the stats, phosphorylated stats, or NF-kappa B, that opens the chromatin, and then the chromatin is accessible for factors that are stem cell factors, transcription factors that were there all the time, but they couldn't access the chromatin until it was opened by these inflammation transcription factors. And then they bind. And now when the inflammation subsides, now these stem cell factors are bound to the DNA and keep the chromatin open so that the next time inflammation is sensed, now the response is much faster. And so this is the model that we're working on, and we're currently using a whole variety of different high throughput technology approaches to be able to identify which are these transcription factors, who's involved, and do they bind DNA, and can this account for the memory that we see? And I think it'll be very interesting to know, because if we can figure this out, we might be able to figure out how to intercept the memory as opposed to treating with immunosuppressive drugs to treat these patients. There's a lot of different questions that we still have not addressed. Do stem cells harbor memories of different experiences? Is epigenetic memory cumulative? How does memory impact aging? Perhaps if memory is cumulative, that could explain why COVID-19, which is a brand new virus, created a far more robust cytokine storm in aging individuals if those individuals had accumulated inflammatory memory before. And remember, the first assault and the second assault need not necessarily be the same. What happens in a world where the secondary assaults are on the rise and varied as they are in our world? Does memory contribute to cancer? We know that some inflammatory disorders are associated with an increased risk of cancer, and we know that mice that can heal their wounds faster are also more susceptible to cancer. Really, at the heart of this is immune cell stem cell interactions, and we're learning more and more that this is a two-way street, central for skin epithelium at the interface between the environment and the body to sustain itself as does the epidermis, the intestine, the linings of the lung, etc. So finally, I'll just touch upon, in closing, our interest in oncogenic stress, because this is another type of stress. We know that as stem cells acquire mutations that will ultimately lead to cancer, stem cells tend to be long-lived, and so they are good vehicles, unfortunately, for acquiring mutations that will lead to cancer, and we're very interested in that progression. We've particularly been interested in squamous cell carcinomas. These account for the 6th most deadly worldwide, the 2nd most common cancers, and they affect a whole variety of different epithelial tissues that I've listed here. I just mentioned that patients that are placed on immunosuppressive drugs or on BRAF inhibitors for something like another cancer like melanoma show an increased risk of metastatic skin cancers specifically. And that incidence of metastatic skin squamous cell carcinoma is predicted currently to be more than doubled in the next 10 years. So we know where the stem cells are, as I've described. We now know where the tumor initiating cells that are propagating the squamous cell carcinoma reside, and they too reside at the interface between the epithelium and the stroma. But now the stroma is very different. There are immune cells, there are different cancer-activated fibroblasts, there are blood vessels. Wherever a blood vessel comes up next to the tumor, we learned that its perivasculature, some of the immune cells secrete TGF-beta, and that can impact the tumor-initiating stem cells at the tumor-stroma interface. And so we generated mice that can sense when the developing tumor cells are receiving a TGF-beta signal and what happens. And I won't go into the genetics, but we could identify then the cells receiving a TGF-beta signal by them turning fluorescently red, and we could turn them fluorescently green if we wanted to track what they're doing and where did they go. We showed that they're slower cycling than cells that don't receive a TGF-beta signal, but these are the ones that are breaking down the basement membrane of the cancer, and they're invading, and we think they are going into the blood vessels because the blood vessels are right there and stimulating this response. So we've also then looked at whether or not this matters with regards to therapy. We've used both cisplatin therapy, which is a chemotherapy, and within a matter of days, the cisplatin therapy on our mice basically kills all the tumor cells that are shown in blue. The red cells are those tumor initiating cells that are receiving the TGF-beta signal, and they're refractory. And if we mark them green and watch what happens to them over time after the cisplatin treatment, they are the ones that are responsible for giving rise to the cancer. And so the tumor initiating cells are important, and they are accounting for cancer relapse, we think, after chemotherapy. We also think that the same is true, but a different mechanism, for immunotherapy. We developed an immunotherapy mouse for our treatments, and within 1 week after treating our mice with immunotherapy to attack the tumor, what we find is that now the tumor initiating cells, the TGF-beta responding cells, go from about 2% of the total tumor to almost a quarter of the total tumor. So again, they're refractory. And if we lineage trace them, this time using a red tracer, we can find that again, the tumor initiating cells that are TGF-beta responsive are responsible for evading the immune system. So where and when along the path to malignancy does stem cell immune cell crosstalk go awry? How does it lead to taking aberrant stem cells that could normally be cleared by the immune system, now to be recognized by the stem cells, but basically evade the immune response and survive. So I think these are very interesting and important questions. And I'll just finish then by saying that often in science, we're looking for something, and no matter how hard we look, we just can't see it. And sometimes if you step back a little bit, you'll see things that you cannot even imagine right now. And I think that's the excitement of science and biomedicine in this day and age. And I really encourage you, whether you're a physician scientist or whether you're a basic scientist, to consider these kinds of important questions for your research. So this is the group of my laboratory right now, and it's a very diverse group. There are several different Canadians, so I encourage you all to think about the United States for a postdoctoral fellowship. But there's a really international group of students from all around the world, and together we have a passion for the science that we do. I'll stop there and take questions.
Thank you very much for your fascinating talk and also stimulating research. So the audience, please use the Slido link that was provided in the YouTube description email that you received, and also the participant code to send your question, and then I will pose them to Dr. Fuchs. Meanwhile, while we are receiving the questions, so I have a couple of questions from you. So it was very fascinating to know the interactions between immune cells and skin stem cells in terms of basically injury and healing process, together. We kind of have the same principles between neural stem cells in the brain and the spinal cord and immune cells, resident immune cells, and also infiltrating immune cells. So you mainly touched based on adaptive immune response. I was wondering if innate immune cells, such as macrophages, neutrophil, or dendritic cells, they are participating in this crosstalk.
Yes, so thank you very much for the question. Indeed, they are, and in fact, the initial discovery of a memory, which is referred to by the immunologists as trained immunity, was discovered in macrophages. Cultured macrophages from the circulation that had a lifetime of about 6 days, so a short memory, just by the fact that the cells died quickly. But nonetheless, when they looked at the epigenetic changes in response to inflammation that macrophages underwent, that they resolved at different rates. And so that was the first concept that memory might be harbored in chromatin, and that the memory was not specific, which I think is a very fundamentally important aspect about this kind of memory. It was then demonstrated that longer-lived macrophages from the lung have this kind of memory, and it was also demonstrated that natural killer cells have memory. And it was about at that juncture that we got involved in this question of memory in the skin. And the surprise came when this is not just a privilege of the immune system, but it is probably a phenomenon that exists not only in epithelial stem cells, but in a whole variety of different tissues. And in fact, we now know that hematopoietic stem cells can harbor a memory as well. So that goes into long-lived stem cells of the immune system. But I think it's this phenomenon, and I should point out that just recently dendritic cells were shown to probably have a memory as well. So it's going to be very interesting to dissect out the crosstalk, because the stem cells are interacting with the macrophages. They're interacting with the innate immune cells, and they're interacting with dendritic cells. And so thinking about on the long term of how each of these different cell types bears a memory, how do they influence each other? I think it's going to be very interesting for the future. And I'll just add one more quick point about the neuroinflammation, because there are a whole variety of neuroinflammatory disorders. And again, there's much less known about the inflammatory memory there, but Samantha Larson, who is my graduate student, is now postdocing to really look at that kind of question of inflammatory memory in the in neural stem cells. I think it will be it's an exciting area for the future.
Yeah, I agree. Thank you so much. So now we have a few questions. So I will read them for you. The number one question is basically everyone is complimenting your research, but so the number one is that, do you think this immunity evasion you see can explain why mesenchymal stem cells are inherently immune invisible?
Yeah, so good question. And some of these things are just starting to unfold. We studied for the tumor initiating stem cells that they evade the immune system. So the classical way in which tumor cells evade the immune system is that people have uncovered the exciting area of PD-L1 expression. If you express a factor on the surface of the tumor cell that normally interacts with immune cells and tells cytotoxic T cells to dampen down their response or exhaust their response, that turns out to be a mechanism that tumor cells hijack. We demonstrated that for the TGF-beta responding cells, that while some squamous cell carcinomas have PD-L1, and PD-L1 therapy has been used in cutaneous squamous cell carcinomas, that there is a related family member called CD80, which also is used by immune cells that interacts with CTLA4, which has also been used in immunotherapy, that turns out to be important and functional. And so we can genetically remove CD80 in that case, and the tumor stem cells are attacked by the immune system. So now is this question of the memory and whether or not they have the ability to dampen down other types of immune attack molecules. And one good example might be the MHCs, because all the cells of our body have to express those to say I'm self versus foreign. And yet in terms of evading the immune system, if they dampen those down, they could do so. And there are a couple examples, one in intestinal stem cells where that happens. And I just point out that stem cells in general tend to have relatively low levels of protein synthesis. And of course, as soon as you have low levels of protein synthesis, you're going to dampen down some of those factors as well. So mesenchymal stem cells, I don't know, but I think it's a fascinating area to study.
Thank you. There is another question asking, have you looked at other epigenetic changes such as DNA methylation or histone modifications to explain the persistent chromatin structure in skin stem cells?
Yeah, I love the question, and we're doing those experiments as we speak. What we know from macrophages is that there are several histone modifications. Actually, the one primary one is H3K4 monomethylation, which appears to be associated with chromatin in the interim. So even though genes are not, the inflammatory associated genes are not expressed, transcribed in the interim, that there are still these histone marks and they correlate with the chromatin accessibility marks that are there. So we think that it's a combination of the two. One of the things about the DNA methylation that I think is so fascinating is that if it's a histone mark or a transcription factor, if it's a histone mark, then, and that's what was really causing the open chromatin, then every time the cell divided, you would eventually lose the memory. But if it's DNA methylation mark, and we now know that DNA methylation and histone methylation can go hand in hand, in fact, in some cases interact with one another, that now you're talking about a stable propagating force. So it's going to be important to see how that unfolds, and we're currently doing those exact experiments. So thanks.
Thank you. There is another question asking, do you think your findings can be common to other epithelial cancers such as breast cancer?
With regards to breast cancer, yeah, I think it's definitely worthwhile looking at. With regards to the lung squamous cell carcinomas have and adenocarcinomas have many of the same transcription factors that are squamous cell carcinomas of the skin have, and they also respond to TGF-beta signaling in an important way. So I think this is certainly going to be a broadly applicable type of a mechanism. We know that in breast cancer, for instance, we know that those cells can be TGF-beta responsive as well. So TGF-beta, initially known as tumor growth factor beta, was associated with tumors. I think one of the things that was surprising to us is that it's long been associated both as a tumor suppressor and a tumor promoter. And we showed that TGF-beta in the skin and in these developing tumors plays both roles, and it plays both roles simultaneously. So it's really heterogeneity within the tumor environment that is responsible for creating this activity. There are regions that are TGF-beta devoid, and those cells are growing, those tumor initiating cells are growing at very, very fast and producing the bulk of the tumor. But they're sensitive to cisplatin and immunotherapy, whereas there's these other cells that are receiving a TGF-beta signal and they're slower cycling, but they're the ones that are invasive, invading and undergoing an EMT. So all of this is happening as the tumor is developing, receiving this heterogeneity in the tumor microenvironment coming from blood vessels, which of course are associated with all of the epithelial cancers.
Thank you so much. And there are many other questions, but for the interest of time and to be on the schedule, we can perhaps, look at them at the end of the symposium. I would like to take the opportunity on behalf of myself and the audience to thank you for your fascinating talk today, and also for your overall contributions to the field of regenerative medicine. Thank you so much.
Thank you very much.
Oh, unmuted now.
There we go.
Thanks, Dr. Fuchs. It was really inspiring and a strong start to the day. So I'm glad that we are sticking to time. I'm glad that Slido is lighting up. I encourage that. We'll try to get all your questions posed, if not now, then later in the town hall. So it's my pleasure now to welcome Dr. Meghan Rastegar in the Department of Biochemistry and Medical Genetics and a scientist with the Children's Hospital Research Institute of Manitoba to introduce our next speaker. Meghan.
Thank you, Jeff. It is with my great pleasure and honor to welcome Dr. Huda Zoghbi to our Gairdner Symposium. Dr. Zoghbi is a professor of pediatrics, molecular and human genetics, neurology and neuroscience at the Baylor College of Medicine. She is also an investigator at the Hughes Medical Institute and the director of the John and Dan Duncan Neurological Research Institute, NRI, at the Texas Children's Hospital. Dr. Zoghbi is a world-leading expert and a pioneer researcher in neurodevelopmental and neurological disorders. She is probably best known for her pioneer work in the discovery of MECP2 being the underlying genetic basis of Rett syndrome. This was published in her Nature Genetics paper in 1999. This paper has been cited over 5,000 times so far, and this is among the impressive number of over 72,000 citations of her. Dr. Zoghbi's research has been highly impactful, and her lab has published original research papers in, reputable journals such as Cell, Science, Molecular Psychiatry, Nature, Nature Family, that included Nature Genetics, Nature Neuroscience, etc. I have known Dr. Huda Zoghbi's research since I started working with Rett syndrome and MECP2 isoforms over 15 years ago in sick kids in Toronto. And I have had the pleasure of meeting her in person a few times and started a collaboration with one of her postdocs that led to a joint publication in 2018. Dr. Zoghbi completed her medical degree at the Meharry Medical College in 1979, and she joined Texas Children's Hospital at the Baylor College of Medicine as a pediatric resident. From 1982 to 1985, she was a postdoc in pediatric neurology at the Baylor College of Medicine. In 1983, Dr. Zoghbi became interested into Rett syndrome by Ben Hager's account in Annals of Neurology. She became then interested on the genetic basis of Rett syndrome and completed a postdoc based on this interest on molecular genetics. In 1988, Dr. Zoghbi joined the Department of Pediatrics at the Baylor College of Medicine as an assistant professor. Soon she was promoted to associate professor in 1991, and then she was promoted to full professor in 1994. Quite impressive. During her career, Dr. Zoghbi has been the recipient of numerous awards and recognitions that only over the last few years, this included the Canada Gairdner International Award and Breakthrough Prize in Life Sciences in 2017, the Ross Prize in Molecular Medicine in 2018, the Victor McKusick Leadership Award from the American Society of Human Genetics in 2019, and she was nominated for the Nobel Prize in Medicine last year in 2020. So without further ado, I invite you all to Dr. Huda Zoghbi's lecture on molecular and neurological studies in Rett syndrome and other MECP2 disorders. Thank you.
Thank you so much, Mojian, for your very, very kind and humbling introduction. Some nominations I didn't know about, so you surprised me with that. But I really appreciate the kind introduction, and it's really an honor to be part of the Gairdner Symposium today. I must say, the Canada Gairdner Prize was one of the most rewarding experiences. Visiting through Canada and getting to know various institutions was very special. And I'm also happy to say that I'm very proud that quite a few of my trainees are in Canada now. So 5 in total, which is wonderful. And I hope this will continue. And I hope I'll get some of the also Canadian students to train in our lab. So I'm going to share with you some of the new research updates on Rett syndrome. And let me go ahead and just share my screen and tell you about some of our more recent work.
there's a lot I could cover about Rett syndrome, but for today's talk, what I decided to do is really just share with you some of the things we learned from studying the neurobiological aspect of this disorder and a little bit molecular, and how this is helping us understand perhaps some vulnerability to neuropsychiatric diseases, and eventually bringing us also to some therapeutic options. So as you heard, my encounter with a patient of Rett syndrome was what really led me into science. And it is this clinical course. People say, why Rett syndrome? I saw so many children with neurological disorders, but why did Rett syndrome capture my attention? I'm going to show you 3 slides, which you will understand why it did. This is Ashley, who I saw at 5 years of age after the whole spectrum of phenotypes of Rett manifested in her. But here you see her as a young infant, baby, where she's holding a book and reading the book and using her hands beautifully. Ashley was able to sing nursery rhymes and do everything a little girl would do. Here you see her riding a rocking horse, using her hand to hold on very effectively. But all of that, after 2 years of age, started to gradually go away, and her hand use was replaced by hand wringing, and all the words that she learned disappear, and her balance deteriorated, and she started developing autistic features and autonomic features, and her gait was abnormal, and eventually seizures. So what really intrigued me is, why was she born healthy? Why did she do well for 2 years and then regressed? But in spite of that regression, there was no neurodegeneration. So what is the process? And reading the paper by Hagberg et al., where all the 35 girls had a similar course, really intrigued me. And finally, a serendipitous event, seeing another girl a week later in the clinic with cerebral palsy. That was the diagnosis, cerebral palsy, but then I saw her clinical course, and she had Rett syndrome, really sealed my fate with Rett syndrome, and decided to go into science to find the Rett syndrome gene. And just for the young student in the audience, I want to highlight that Rett syndrome is a sporadic disease, which means it's one in a family, which means with the technology we had in 1983, 85, when I'm collecting samples on Rett syndrome girls, we couldn't really find the gene because you basically could not map it. And the only thing that helped me is that I formulated the hypothesis that this must be on the X chromosome because it's always girls. And even though it seems to be a new mutation in every family, the fact that it's girls, maybe that will help us. And indeed, it was simply through consistency, narrowing regions on the X chromosome when we find an occasional half-sister pair that were affected, led us to discover the gene in 1999 to encode methyl CpG binding protein 2. Now, this protein was first discovered by Adrian Bird in 1992 as a protein that binds methylated cytosines, and typically when followed by guanine, hence its name, methyl CpG binding protein 2. But since then, I'm just going to summarize here quite a few data. We learned that it also can bind non-CpG methylated cytosine. Hence, the H could be any nucleotide. Most commonly, it's an A, adenine, but it can be any of those. And it's enriched in the postnatal. This mark is quite enriched in the postnatal brain, which means it increases as the brain matures, and that parallels the abundance of MECP2. And to us, that also parallels the delay in onset of symptom. But that's not the whole story why we have delayed onset. I'll come back to that. More recently, we found that this mark is really critical. Quite a bit of the gene expression changes mediated that result from loss of this protein is mediated by this mark. And also Adrian Bird showed that other methyl binding domain proteins that don't read this mark cannot rescue the Rett syndrome phenotype. So altogether, we know now that this MCH mark is important. Going back to the Rett syndrome phenotype, human patients teach us a lot about the full phenotypic spectrum of a disorder. So after discovering the gene, we learned that majority of the cases, over 95% of the cases with Rett syndrome, have mutations in this gene. Now, I told you this is on the X chromosome, which means the girls are mosaic. Half of their cells lack the normal allele and half have a normal allele. And just having 50% of your cells with a normal allele is not enough, will lead to all these features of the syndrome, as you see here. What we also learned that there are actually males with mutations in this gene, but these males, because they have a single X chromosome, they have a very severe phenotype when the mutation totally inactivate the gene. And you'll see most of these males die, unfortunately, by their second birthday. What we've learned in more recent years is that the phenotypic spectrum is much broader. And we learned that some mutations that are actually milder, that partially inactivate the protein. So you still make a protein, but that protein, it's in a male in every cell, but it doesn't function as robustly. Now you begin to see more psychiatric phenotypes layered on mild learning disability. So those children are not as severe as the null males or as Rett syndrome girls. They have mild learning disability, and they can have any of these neuropsychiatric features, either autism or anxiety and OCD, or ADHD and tremors, bipolar disorder, juvenile schizophrenia. And these features are in different colors on this slide because they don't occur in the same patients, but it's a combination of a psychiatric feature on top of a mild learning disability, and typically tremors are common. So recognizing this is quite important because it tells us we haven't really seen the full spectrum of the phenotypes yet. The other side of the coin, we learn that if we increase this protein, we're going to also get progressive neurological disorder. And this is a situation where it first appears in mice. As a positive control experiment for a gene therapy experiment, we added the human gene because we figured we'll add the whole genomic locus and then breed it to the null mice. That'll be our positive control. Then we can try to use vectors for gene therapy. Well, when doing this positive control experiment, first you put that human gene using its own regulatory elements in the mouse on top of the mouse endogenous gene. So this is a transgenic. And to our surprise, doubling the level of MECP2 resulted in progressive neurological phenotypes and autism-like features and stereotypies, epilepsy, and even early death in these mice. By 1 year, they died. So this immediately alerted us that increasing this protein could also be bad. And we suspected that there may be humans with duplication spanning this gene. And indeed, within a year, Linda Vanesh discovered MECP2 duplication syndrome in boys, where these boys have duplication spanning the MECP2 locus, and they have all the features we've seen in the mice, including respiratory infections, and of course, early death. Typically by the third decade, many of these boys pass away. So this told us that the dosage of this protein really matters. We also learned that the duplication is causing disease by gain of the function of the protein. Neurologically, they looked similar, but physiologically, at the neuronal level, they were opposite. Loss of the protein makes too few synapses, gain too many synapses, and gene expression changes were opposite. The null mice had thousands of genes changing, and the duplication had those same genes changing, but in an opposite direction. So it is really a gain of function of the protein and a gain of the normal function. So in summary then, from all the human and mouse studies, we learned that the levels of this protein matter, that total loss is fatal in males, loss in females, which is mosaic, is severe. We even learned from animal studies, if you decrease it by 50% if in every cell, you get a moderate phenotype. And I shared with you some of the neuropsychiatric phenotypes that we see with milder mutations. This is an example of such mild mutation. And on the other side, we learned that doubling the protein causes severe neurological problem, and triplication cause even more severe disease and earlier fatality, both in humans and mice. And I would say that all the features we see in the humans are replicated in the mice. So in this case, and of course, I shared with you that the duplication mouse predicted a human disorder. So in this case, the mouse models are really genetically and phenotypically accurate representation of what we see in the human disease. But having this sensitivity to MACP2 level, we rationalize that probably there are multiple layers of regulation to keep this protein at precisely the level you want it at. And that inspired us to look into transcriptional regulation of this gene. And to do this, a graduate student in my lab, Yin-Yang Shah, collaborated with a colleague of ours at Baylor, Josh White, who was doing ATAC studies. These are studies to look at open chromatin regions and regions that may be accessible to transcription factors. And he did that in the early embryonic brain, in the early postnatal brain, and in the adult brain. And he did it in peripheral tissue. So he was doing these data sets as part of another study, and we collaborated together by looking at what regions are accessible for regulation, transcriptional regulation in the adult brain, because that's when MACP2 levels are the highest, and they increase over time to see them the highest, or these regions most active, if you will, in the adult brain. And of those, can we find conservation in the human data? So I'm sharing with you here, at least the adult brain data, because that's when the MACP2 levels we know absolutely are mature. And you'll see several peaks that were identified. The only previously known peak was the promoter region. This was discovered by Uta Franca many years ago. And we knew that deleting this region totally abolishes transcription. So we knew about this one. But these additional new peaks shown here, peak 5, peak 6, and you see some of them are most prominent in the adult brain. Some of them occurring within large introns. So this is the first exon. This is alternatively spliced exon that Meghan contributed to its discovery and characterization. And these are the remaining last 2 exon. And you'll see many of the peaks are in introns or upstream. So we wanted to see, do they have consequences on the RNA and protein level? And to do this, Yin-Yang created mice that deleted each one of these peaks. We did not do the promoter because we knew it's going to kill gene expression, but we wanted to see these additional peaks. What do they do? And what you'll see here in the brain tissue, that manipulating these regions does actually affect protein level. This is the wild type animals, and you see the amount of MACP2. This is the GAPDH control. And you'll see knockout of peak 1 has a slight increase, but knockout of peak 2 significantly decreases the level of the protein, almost by 30 to 40%. And the other 2 peaks have very minimal, if any, effect. But knockout of peak 6, you see now quite an increase in MACP2, almost 70% increase in MACP2. So because these 2 peaks, peak 6 and peak 2, had the biggest effect on the level of the protein, we wanted to see if they're conserved in human brain. And for that, we looked at ATAC-seq data from Jeremy Nathan's lab that he's published on the human brain, and we found that these 2 are indeed conserved. So now we have to see if you delete them in human neurons, do you affect protein level? And for that, she created engineered eye neurons that either lack peak 2 or peak 6. And you'll see here again in the wild type neurons, human neurons, the levels of the protein are quite high, but peak 2 knockout results in, again, 40 to 50% reduction in the protein, and deletion of peak 6 increases the protein level. So now we know these 2 peaks regulate the RNA and subsequently the protein level, and we know they're evolutionarily conserved. Is there a consequence for reducing the protein by 30% or increasing it by 50%? And for that, we deeply characterize the animals. And I'm just going to summarize the data for you because we don't have a lot of time to go through all the data. I want to remind you that you have to have the protein at 100% for normality. And if you have mosaic or you reduce the protein by 50%, we see all the features in these big blue circles. However, if we only reduce the protein by 30%, we get a subset of the features. As you'll see here, we get mostly neuropsychiatric features, hyperactivity, anxiety, and social deficit. This is going along with what we learned from the human data, where a mild mutation also gives you a subset. There's also some learning disability. So this is probably milder than that mutation. Hence, we only see hyperactivity, anxiety, and social deficits. On the other side, I mentioned to you that our MECP2 duplication mice had a variety of neurological features shown here. What you now found, the deletion of peak 6 that result in 50% increase. Now we get a subset of these features. Again, quite a bit of psychiatric phenotype, but here we see some learning and memory deficit and a little bit of hyperactivity. So basically, we see that you get a subset of the phenotypes, mostly along the neuropsychiatric or cognitive phenotypes, but we're beginning to see with 50% increase a little bit of motor phenotype, hyperactivity, but not all the rest of the major phenotypes, such as major motor deficits and epilepsy. So this allows us to now add to this curve that I shared with you, where a 30% reduction or a 50% increase can cause mild and moderate neuropsychiatric phenotypes. And this is in mice. So to see a neuropsychiatric phenotype where we can only assay for a few things, lead me to really propose that even milder changes, perhaps in the 10 to 15% range in either direction, might actually present with isolated psychiatric phenotype in human that may be harder to see in mice, but probably in a human will manifest, maybe later onset disease as typical of adult psychiatric features. So this highlights the importance of studying these regions as well in genetics, because typically when we sequence this gene, most of the time it's exome sequences, these regions in introns are left behind. So I think now that we know a slight change in or a deletion of these regions can make an organism susceptible for neurological and psychiatric phenotype, it would be really important to sequence them in samples of autism and psychiatric disorders, so they could hopefully be included on the capture sequencing. Now I want to switch to the neurological or neurobiological aspect of our studies. And here I'm going to probably summarize at least 10 years of work in just a few words, telling you that when we wanted to understand the origin of all the features of Rett syndrome, we deleted this gene in various neurons. And to our surprise, we discovered that so many neurons depend on this gene for normal function. And we would get minor phenotypes, typically one or two. Here, obesity, overeating, and stress. Here, motor abnormality. Here, premature death and some autonomic problems, and so on and so forth. Of course, for GABAergic neuron and glutamatergic neurons, we got a multitude of phenotypes that complemented each other. But the reason I wanted to share the results of this study is to tell you that what we learned from all these studies is losing this gene partially disables the neurons, making them function at about 70% of their normal capacity. And all of them are important. So we quickly discovered that if you normalize the gene in inhibitory neurons, you might correct the phenotype for a while, but then the system will crash again because you don't have it in glutamatergic neurons, and vice versa for the glutamatergic system. This tells us you really have to improve the circuit. You cannot just improve one subset of neurotransmitters. You need this gene in every cell because all the cells, a majority of the cells, don't work at full capacity. So to follow up on this, one of the experiments we did to see if we could manipulate the circuit. What would happen if we increase the circuit activity using something that's used in clinics for certain disorders, and that is deep brain stimulation. And we asked if we focus on one circuit, the hippocampal circuits that mediates learning and memory, and did repetitive stimulation using deep brain stimulation, would that improve the learning and memory? And for this, we collaborated with Zhangrong Tang, who leads our in vivo physiology core, and Shuang Hao, who is a postdoc in his lab, who did this work, and took adult Rett syndrome mice and implanted them when they are adult and where symptoms of learning and memory already appear at about 2 months of age. And they received 1 hour of deep brain stimulation using the same parameters used for humans who undergo deep brain stimulation, either for epilepsy or tremors, and of course, Parkinson's disease. And here the fornix was stimulated with the idea that the fornix, which is shown here, will have projections to its neurons will have projections to the hippocampus, and that will increase the excitatory activity and the activity of the neurons in the hippocampus. And we assayed all the hippocampal phenotype, fear conditioning, Morris-Water maze, this is spatial learning, in vivo long-term plasticity, neurogenesis, and gene expression studies in these animals. And I'm just going to summarize, because all of this is published, all of that corrected. So learning and memory corrected, plasticity corrected, the synchrony patterns in the CA1 neurons normalized, neurogenesis increased and normalized, and gene expression changes improved. So this was quite exciting, and we are following up on that to see if stimulating other areas would affect motor function. But we realized that in Rett, there's so many phenotypes, and we were thinking it's going to be really hard to do deep brain stimulation to really correct all the various phenotypes. So we began to explore some other ideas. Is there something we could do that could maybe mimic a little bit deep brain stimulation, but it's less invasive? And for that, we asked, would intensive behavioral training, because you're engaging neurons in activity during behavior training, but if you did it intensely, would that enhance circuit activity and improve the Rett phenotype? And we knew, of course, that many Rett girls undergo physical therapy once their diagnosis is made. So we wanted to ask, is the timing of that therapy important? If we did it before the symptoms occur, would that be helpful? And we knew also from other studies, from Tony Hannan, Wan Yang, and Peter Russo, that enriched environment for the Rett mice has improved their functionality. So we designed an assay to see if early training on a rotating rod, which the Rett animals do poorly on once they're symptomatic, if that will help. And in this case, we did the training at 8 weeks of age, because the mice become symptomatic at 12 weeks, and they become unable to be on the rod at 24 weeks. So if we were to test naive animals at 24 weeks, they immediately fall off. So what we did is we started the training at 8 weeks, and we did it every 2 weeks. They had training sessions for a total of 18 sessions of training before we test them at 6 months of age. But to be sure that it's not just the number of training, that the timing of training matters, we did the same number of training sessions after symptoms onset. And of course, we have the naive animals. And what you'll see here is that there was a big difference. Here's the naive Rett mice. They quickly fall off the rotor rod. The black bars are the controls. And you'll see that the late training was helpful, but not as helpful as the early training. And when we followed these mice, we actually delayed the onset of symptoms. And even then they were milder by several months, by 5 months. So this is work of Nate Akili, who was an MSTP student in the lab. When we tested these animals on other tests, we did not see benefits. So we were curious, is it just the rotor rod that's going to improve, or could we actually help them with a different type of task? So we turned to learning and memory. And here, if you put the animals in a Morris-Water maze, after 4 days of training, a healthy animal will quickly find the hidden platform using cues on the walls. But unfortunately, the Rett mice can't do that after the 4 sessions of training. So we asked again, in this case, this phenotype happens early at 6 weeks. So we started at 4 weeks and we didn't have much time to train, but that period of time at least was still during the normal period. So we designed the experiment that at least we gave them 2 extra training. That's all we could afford within this short period every month. So they got a total of 12 training sessions, and they got the same sessions after symptoms, and of course the naive animals. And the data are really beautiful in that what we learned is that naive Rett animals simply after 4 days can't find the platform, whereas wild type quickly will find it. Rett animals trained late still cannot find the platform, but wild type animals trained early or trained late, as well as in this brown line here, the Rett animals trained early now can find the platform as quickly as the wild type animal. And importantly, they can also remember where it was if we took the platform away. They will search that quadrant and the target area more efficiently. So you'll see here, naive Rett animals, it's just changed, it's 25%, it could be any quadrant, but the wild type animals search the target quadrant more often. The late trained animals, similar to chance, but now the early trained animal search a quadrant more, and particularly they will spend more time where the platform used to be, so they'll have much more platform crossing than the naive and the late trained animals. But what was interesting, again, they only did great on this task. They didn't do well on other hippocampal tasks. And this led us here, for example, contextual fear conditioning, they didn't do well on that. So this really led us to suspect that perhaps we're only activating the task-specific neurons, and that's why we're seeing the benefit. But is that reality? I mean, is it really about those neurons engaged in the task? And to do that, we have to capture those neurons, we have to label them, and we can manipulate them. We can silence them, we can activate them. And that's what we did. We used a mouse line called the Fuchs trap mouse, where any neuron that's activated, so when a neuron is involved in a task, it's activated. And in this case, when it's activated, it's gonna turn on a recombinase that will allow to remove a stop signal and label a reporter. And we give tamoxifen just when we put the animals in the task, so we know that the timing is just precise. And you'll see now this neuron labeled, and you can now do anything you like with this. You can label it, or you can express in it, again, a Cre-dependent receptor. And just to show you here, if you take mice and you handle them, not much activity, but if you take mice, put them in the Morris-Water maze, you see all the hippocampal neuron and cortical neuron engaged labeled. So now that we can manipulate these cells specifically, we decided to express these designer receptors that are activated by designer drugs such as CNO, that this normally is not activated in our body, but if you have the receptor, it will activate. And there are 2 forms of these receptors. You can either express a receptor to silence these neurons or to activate them. So in this case, we wanted to put the animal through our regular training and see, do they learn? And of course, they're gonna learn. We can give them CNO, but because there's no receptors, they will still learn. These animals have M-cherry. But if we express the silencing receptor, we wanted to see, after all the training, if we just silent only these task-specific neurons, because this is a Cre-dependent and the Cre is under control of activated neurons, what will happen to learning and memory? And the data are very clear. You'll see if the animals have been trained and only had M-cherry, again, this is, if you will, a replication of our experiment that early training, the animals will perform similar to wild type. You see here, they learn quite a bit. But if you silence the neuron before testing them, you give them all the training, they're trained just as much as these, but we now silence the task-specific neurons, the benefit is gone. So then we asked, is it really because we're activating the neuron? Is putting them in the pool time and time and time again is resulting in activation of these neurons? And would simple activation without training work? So in this case, we put the animal in the pool once just to activate these sets of neurons and bring them back to their cage and give them the drug either with M-cherry, so the drug is not going to do anything. So these animals really going to forget. They're not going to be well trained on this task. Or give them the activating receptors. So those neurons that got activated in the Morris water maze, now they're activated, and the same time they express the activating receptor. And every time we give them the drug, only the neurons are activated, but they're not seeing the pool. But those neurons that got activated in the pool now is being repetitively activated, and we test them again. And this is what we see. If you don't train the wild type animals and you simply give them CNO, just like the Rett animals, they're not going to do well because they need at least 4 repeated sessions of training. But if you give the activated receptors after just 1 session of activating these neurons, now they do very well. And they do the platform crossing again very similarly. So stimulating these neurons is really activating. So I'm just going to summarize what I shared with you. We learned that pre-symptomatic training improves motor and memory deficits in the Rett mice in a task-specific manner, that discontinued training delays the onset of the phenotype, and that the task-specific neurons mediate the beneficial effect. I didn't show you the data today because I knew we would be short in time. We improved the morphology and the physiology of these neurons. And this has implications for Rett syndrome, because in Rett syndrome, I told you that the disease onset is usually typically after 18 to 24 months, and the majority of the girls are diagnosed by 3 years of age. So we have this whole period of missed opportunity that we might be able to do some intensive training on various tasks. And if we do that and test that in clinical trials, if there was newborn screening, we might actually delay the onset of Rett syndrome with this intervention. And we might even keep them longer functioning where they could be responsive to any more permanent therapy that will come down the line. And just in the last couple of minutes, I'm just going to tell you the other side of the coin, which is the duplication story, where we have the animals that replicate the features of the disease. And here we asked if we reduce the protein using antisense oligonucleotide in adult mice, would we reverse the symptoms, and could this be used as a therapy? And for this, we collaborated with Ionis Pharmaceuticals. And I'm just going to summarize very briefly. Here's an example of the transgenic animals being hypoactive. And you see these animals improve their activity, their rearing, their entry to the center. You'll see here the same animal on ASO improved similar to wild type animals. And this was work of Hezi Steinberg. Then we waited till the animals were 7 to 8 months old, and they're having seizures every day, and they're dying actually from seizures. We can confirm them by EEG and clinically. And if we treat with ASO, we normalize the EEG and suppress the seizure activity clinically. So that was adding the human gene on top of the mouse gene, and we targeted the human gene. But in human, we have 2 identical genes, and I told you how important the dosage of MACP2. So we created a new mouse model with 2 human genes and no mouse genes, and asked, can we titrate the drug as a proof of concept first step for clinical studies? And the answer is yes. Based on the dose you give, you can titrate this drug just as you would titrate a blood pressure medication. And the next question we asked is, how soon after we give the drug does the RNA goes down and the protein goes down? And what was interesting, we noticed that the RNA goes down very quickly within 1 week, and the protein really goes down 2 to 3 weeks later. So there's a lag time, 1 week lag time between the RNA and the protein. But what was most interesting for us is, when does the clinical benefit appear? And I summarize all the data here. You'll see these are adult mice where we first give them the antisense oligonucleotide in the ventricle as an injection. and you'll see early normalization of the RNA, followed by the protein, followed by many of the gene expression changes I told you that happen. But what was interesting, behavioral changes didn't happen till about 9 weeks of age. There's almost a 9-week lag time between therapy and benefit. But what's important is we have a window of opportunity to monitor the dose. We give a dose and we can follow perhaps some of the targets, some of the molecular changes, and that will help us adjust the second dose, and if necessary, give an antisense to the antisense if we happen to lower, might be too much. So these are the proof of concept data now that led to clinical trial readiness studies that are ongoing to define these early biomarkers that we wish to use once hopefully we can move to clinical trial. So with that, I hope I convinced you that both loss and gain of this gene causes a variety of neurological disorders. But what's interesting is the milder mutation causes psychiatric phenotype, and the antisense oligos can be used in the future, hopefully, for treatment of MACB2 duplication. And we have a time lag that hopefully allow us to monitor the therapeutic window and levels. And the pre-symptomatic training could be beneficial in Rett syndrome. And hopefully, with time, we'll move into newborn screening so that proper trials could be done for that. And this is our group. Once we all got vaccinated, we were able to have a picnic in the park. They're all wonderful. And I will just acknowledge there are many people who contributed to the conditional knockout study, but just a few people to acknowledge. Ruthie Amir discovered the gene. She was tenacious. Always will acknowledge her. Nate did the early training studies in Yinyao, found the regulatory elements, and she did the proof of concept, most recent ASO studies. Wei Wang helped with the physiology of the early training, and our collaborator, John Rong, and a collaborator at Ionis, and Josh White. Thank you very much.
Okay, thank you so much, Dr. Huda Zoghbi, for this fascinating and exciting talk. So we're gonna take a look at the questions. I think there are several questions that have been posed. So we have one question. Okay, it's interesting that both the MECP2 promoter region and PIK7 are open to enhancers and repressors in young mice. I have two questions related to this. First one is, have the MECP2 enhancers and repressors been characterized?
Excellent questions. What binds these peaks? We're beginning to do that. We've identified a binding site for CTCF in peak 6 that we believe may be playing a repressor role. But we also identified next to that site in the human, at least brain, TCF7-like 2, a putative transcription factor that binds this region. And interestingly, loss of one allele of this gene causes autism and intellectual disability. So we are now performing chip studies to see if this binding really is there, map the precise binding of that regions. And over the next few months, we are following up to identify what also binds to PEAK2, to identify the factors that bind them, because they will be candidates for autism genes as well.
Thank you. And so now, is there evidence of a potential molecular switch that promotes MECP2 expression later in life?
That's interesting. The question is, why is it there are more higher levels in the adult brain? And is there something, we believe it's going to be one of those regulatory regions. And we hope that once we identify the transcription factor that binds those regions, that will tell us at least this may drive the timing of the expression. So the answer is yes, but we have not identified it yet.
Okay, thank you. There are some, I guess, compliments about the talk. Everybody seems to be enjoying the talk. There's another question. Does levels of MECP2 proteins only affect neurons in Rett syndrome, or it impacts glial cells and oligodendrocytes?
Excellent question. At this time, most of the data are on neurons. The protein is almost 7-fold higher in neurons than it is in any type of glial cell. And as I mentioned to you, deletion in neurons specifically can recapitulate all the phenotypes of the disorder. But that being said, it probably plays some roles in astrocytes because Gail Mendel has shown that astrocytes are important for supporting the neurons, the Rett neurons in culture. Wild-type astrocytes do much better than Rett astrocytes. And then when she did a conditional rescue, she did improve the breathing phenotype by rescuing the protein in astrocytes. There was not as extensive characterization, so we can't tell about long-term survival and long-term neurological and other. So there's possibly some role for astrocytes and glia, but it is not the driving force of the disease as much as the neurons.
Okay, thank you so much, Dr. Zoghbi. I think it's time for a break. For the sake of time, I would invite everybody to join us in the afternoon for the Q&A session. Thank you so much. Over to you, Jeff.
Thank you, and thank you for a great lecture there. So we're going to have a short break, 15 minutes. We'll start at 1 o'clock sharp. Keep the momentum going and the enthusiasm high. So we'll see you at 1 o'clock. Thank you. Welcome back, everyone. It's my pleasure to introduce Dr. Lee Murphy, Professor of Biochemistry and Medical Genetics and former Scientific Director of the Cancer Care Manitoba Research Institute, to introduce our first speaker of the afternoon, one of our own, Dr. Meghan Azad. Lee?
Thanks, Jeff. Good afternoon, everyone. It's a real pleasure to introduce Meghan today. I have known her since she was an honors student in my lab back in 2003. At that time, it was immediately obvious that in addition to being extremely smart, she was also very hardworking, articulate, dedicated, and extraordinarily organized. I actually picked up quite a few tips from Meghan, reorganizational skills, and I thank you for that, Meghan. Since then, I have followed with delight, but no surprise, Meghan building a very successful research and academic career. She is currently an associate professor in pediatrics and child health here at the University of Manitoba. She's a scientist in the Children's Hospital Research Institute, and she co-directs the Manitoba Interdisciplinary Lactation Center, which includes the Human Milk Biobank. Along her career pathway, she has won numerous prestigious national and international career awards, and she currently holds a Tier 2 Canadian Research Chair in the developmental origins of chronic disease, with a focus on human milk composition and the microbiome, the infant microbiome, breastfeeding, maternal nutrition, and their relationships to long-term health outcomes. Since setting up her own research program, she has been continuously well-funded, holding numerous research grants as a PI or a co-PI, including funds from the CSHRF, the Bill and Melinda Gates Foundation, and the Canadian Institutes for Advanced Research. True to her own interdisciplinary background in molecular biology and epidemiology, she leads several interdisciplinary teams, such as the Child COVID-19 Study and the International Milk Composition Consortium. Given all these substantial resources and her very hard work, as you might expect, she also has an impressive publication record for this stage of her career. With 98 and counting peer-reviewed papers, many of them in high-impact journals such as Cell, Host, and Microbe, JAMA, Pediatrics, etc. And I think you started your peer-reviewed publication record with a first author publication from the work you did in my lab as an honors student way back then. And she has an enormously long list of scientific and non-scientific interviews and presentations, both locally, nationally, and internationally. So I will now invite Meghan to take over and tell us about her exciting research on human milk, the ultimate superfood, and personalized medicine, and some of the research activities at the Manitoba Lactation Center. Welcome, Meghan.
Thank you so much. Just checking that you're hearing me okay and seeing my screen. I'll take that as a yes.
I can, yes.
Great. Thank you so much, Lee, for that really kind introduction. You're right, my very first paper was the one from your lab, so it's extra special to have you introducing me today. Really appreciate the kind words, and also want to thank the Gairdner Foundation for the invitation to speak today. It's a real honor to be presenting amongst the highly accomplished Gairdner laureates that are part of this symposium. So I'm really excited to speak to all of you a little bit about my research on human milk. I want to start with disclosures. So these are my funding sources, for which I'm very grateful. And I'll start with personalized medicine. That was in my title. This is a concept, I mean, it's a buzzword. I think different people, it means different things to different people. I think in a clinical audience, a lot of people think about personalized medicine as, Lee introduced me, we started working together on cancer. So a cancer drug that's personalized to the particular genetic polymorphisms in a tumor, for example. Or if you talk to nutritionists, they might think of it as a personalized exercise regime that's optimized for you and your metabolism or your body type. But when I think of personalized medicine, I think of human milk. And this is a quote that I love by Cesar Victoria, published in The Lancet, which did a special breastfeeding series in 2016. And if you get inspired by the talk today, I'd encourage you to go here as a place to start in terms of the summary of the research and evidence around breastfeeding, not only from a health perspective, but also an economic and public health perspective. So Victoria wrote in this excellent series that human breast milk is not only a perfectly adapted nutritional supply for the infant, but probably the most specific personalized medicine that he or she is likely to receive, given at a time when gene expression is being fine-tuned for life. So I love this quote because it wraps up for me in one sentence a lot of what I do as a researcher. So this idea that the timing is really important. So I'm a DOHAD researcher, DOHAD meaning the developmental origins of health and disease. And that entire field is focused on the idea that what happens very early on in utero or in early infancy can really have a lasting impact on life, because this is when so many of our physiological systems are programmed. So our immune system, our metabolism, and so on. And that's the period of time when breastfeeding occurs. And it also refers to this fact that breast milk isn't just food. Of course, it is food, and it's very important food, the only food that infants are consuming early in life during this critical period of time. But it's so much more than food. It contains many other components, and we'll talk about some of those today. I start out a lot of my talks with this quote, but it's especially fitting today because Cesar Victoria was a recipient of a Gairdner Award in 2017. And I can actually still remember sitting on the couch in the morning, eating my oatmeal like I always do, reading the news, and seeing the announcement of the Gairdner awardees for that year. And scrolling through, there are about 5 or 6 of them each year. And I was, about to close the article. Cesar was listed last, and I almost spit out my oatmeal because it was so exciting to me that a breastfeeding researcher would have won an award of this caliber. And if you've been watching the session today, you heard an introduction about Gairdner. These are known as the mini Nobel Prizes. It's a big deal to win a Gairdner Award. And the fact that breastfeeding was recognized on that level was really exciting to me as a new researcher starting out in that area. So I was really excited, and Cesar Victoria was kind of a hero of mine. He really pioneered a lot of the epidemiology in the 70s and 80s that showed breastfeeding was so important for infant health. There had been a period of time before that when it was thought that infant formula, which was this kind of new scientific substitute for breast milk, was much better. And it was really his work that started the ball rolling of understanding that no, breast milk is personalized in a way that formula never could be. And he not only did research to show that, but also did a lot of work in policy and implementation to find ways to support breastfeeding. So not just stopping at making the discovery, but making an impact with that information. And so I really wanted to meet him, and I reached out to the Gairdner Foundation and said, can I come to the awards? And they said, sure, it's open to anyone. So I flew to Toronto, and I got to meet Cesar Victoria, one of my research heroes, and it was an amazing experience. And I won't go into that story right now, but I know many trainees are watching and participating in the mentorship sessions tomorrow. So if you ask me, I'll tell you the story of how this turned out to be a $5 million moment. And we can discuss that at the sessions tomorrow. So with that, I'll sort of move on to the meat of my talk, which is the research and evidence around breastfeeding and human milk and why I'm so excited about it. So I talked about breast milk as personalized medicine. This is kind of one buzzword. We also talk about breastfeeding sometimes as a baby's first vaccine. There are actually countries where the vaccine record starts not with the baby's first vaccine shots, but with whether or not the baby got colostrum, because it's that important to immunity early on. Also in the title of my talk, I talk about breast milk as a superfood. So we'll get into this a little bit. Often you hear colostrum, which is the very first milk, referred to as the original liquid gold. And maybe a bit cliche, but I really like this. Making milk as a superpower. This is a pretty awesome thing that mothers are able to do. And it's important to understand how it all works. And that's what my research really focuses on. So starting with this superfood claim. This was a perspective published in Science a number of years ago and framed breast milk as nature's first functional food. And so I really like this, this perspective. And this is another quote from that article that I really love by David Mills, a human milk researcher. He says, "Milk is really a genius fluid that was outrageously understudied. If we could identify the components of human breast milk that are important, then we can understand the wisdom of milk and take advantage of them." So what does he really mean about taking advantage? Well, to put some numbers to this claim, these are global data on child mortality, so mortality of children under 5 years of age globally. And at the Institute for Health Metrics and Evaluation website, I generated this map myself. So they have all sorts of data available and you can play around with it, looking at, for example, cause of death. And so this is a map showing you the proportion of child deaths under 5 years of age per country that are attributable to suboptimal breastfeeding. And these are data from 2013. So you can see that, fortunately, in our part of the world, not too many child deaths are attributable to breastfeeding suboptimization. But in some areas of the world, up to, this is close to 20% in South Africa of childhood deaths under 5 years of age could be traced back to suboptimal breastfeeding. This is a country where if babies need to have formula, that formula is often made with water that is unclean, that might cause infections. The medical care there is not as good as we would have in other places. Infectious disease is a major cause of mortality, especially in infants. And you can see that in other areas of Africa, there are 10 to 15% of deaths. So that same Lancet series that I quoted Cesar Victoria earlier has estimated that improving breastfeeding practices around the world could save over 820,000 lives a year. So these are big stakes when we think about breastfeeding and how we could take advantage of the knowledge of breast milk, even if every baby can't be breastfed, because we know that's not always possible. If we can understand how breastfeeding and how breast milk supports infant health, then this is information we could use to support health in all babies. Now, of course, that's one extreme, childhood mortality. And you can see it's not so much a problem in our part of the world, but that doesn't mean breastfeeding is not important here too. So this is a handout from Manitoba Health, so right in our own backyard, talking about reasons to breastfeed your baby. And so there are many, including that it's better for the environment, it can save you money. And the one I will focus on today is that breastfed babies are healthier. So it's not just about childhood mortality, but thinking in our part of the world, breastfeeding has been associated with a 36% lower risk of sudden infant death syndrome, a 72% reduced risk of respiratory tract infections during infancy, a lower risk of type 1 diabetes, and also a 15 to 30% reduced risk of obesity later on in life, so adulthood or adolescence. SIDS and infections, these are things that happen in infancy, but diabetes and obesity, these are lifelong conditions that can be tied back potentially to breastfeeding. And then one reason that wasn't on that pamphlet, because it's a bit old, is to do with the microbiome. And this is actually how I very first got interested in breastfeeding. So during my postdoc, I worked with a child study, which I'll tell you more about in a few moments, and did some of the first analyses of the infant gut microbiome from that cohort. We published a paper in the Canadian Medical Association Journal in 2013. It's really small here, but it says profiles by mode of delivery and diet. So we found that the microbiome of babies was different depending on whether they were born by C-section or vaginal delivery, and also by whether they were receiving formula or breast milk. At the time I did this paper, a lot of people didn't even know what the microbiome was or why it was important. I hazard to guess that if we were all in a room today and I asked you to raise your hand if you've heard of the microbiome, I think all the hands would go up. It's pretty well known now that these bacteria that live in us and on us are really important. And it's becoming clear that early in life, breastfeeding is really important for shaping that community. So again, I won't tell the story today, but maybe something for the mentorship sessions tomorrow. This paper, which you can see made the cover of CMAJ and actually received an award for Paper of the Year from that journal, was initially rejected and not even reviewed by the journal. So that tells you a little something about perseverance and also the way that science attitudes can change over time. 5 years later, this paper, which I was not involved in, but comes from another birth cohort study, made the cover of Nature. And with a larger sample of infants and more advanced techniques, they replicated what we had seen, which is that breast milk, of all these different factors that might influence the microbiome, was far and away the most important factor shaping the infant microbiome. So that's this bar here. You can see that as we showed, how you were born is important, it's significant, whether or not a mother's obese, whether the baby takes probiotics, all of these are important, but by a landslide, breast milk is the most important. So why is that? And why did I then go on to focus my career on human milk? Well, to understand that, we really have to start looking at the milk itself. And so, you're not intended to read this, but I think this picture shows you very clearly that there are many things in human breast milk that simply aren't in formula. And formula companies are working all the time to make formula better, make it closer to human milk, but it's kind of no competition, and it would be very hard to replicate. And one of the reasons for that is that human milk is dynamic. So it changes. It changes from day to night. So the milk that a mom makes in the morning is different from the milk that same mom makes later in the day. It also varies even within one feeding. So milk at the beginning of a feed is lower in fat, and then the fat content increases towards the end of a feed. And that's thought to help babies understand satiety. And it also varies across lactation. So as the baby grows and develops, the composition of milk changes over the months. And it's also very different among mothers. So different moms are making different compositions of milk. And you can see that all of these things would be very hard to replicate in a one-size-fits-all formula. Milk is also personalized. So we talked about milk being a superfood. As a nutrition, it's personalized. It matches to meet the needs of the infant over the course of lactation. And it's also very specific to the species. So, all mammals make milk. That's one of the defining features of mammals. But if you think about just two species, so cows and humans, a human baby spends a lot of its infancy period sleeping. This is when its brain is developing, whereas a baby calf, if you've ever seen one be born, sort of is born and right away wobbles and gets up and starts moving around, and its brain is pretty much fully formed when it's born. So because of those huge differences in the priorities of a baby human versus a baby calf, the milk composition is really different. So human milk, for example, has much higher levels of fatty acids that are important for brain development. So if you start feeding cow's milk to a human baby, you can see how there would be a mismatch. And then we talked about human milk as a personalized medicine. So milk is not just delivering nutrients, it's delivering optimized immunity to the baby while its own immune system develops. So a baby is born with a very immature immune system, and it's relying on its mother's milk to get things like antibodies to protect it from infection while its own immune system develops. And so this is another picture showing you a similar message, but you'll be able to read the words, which is that formula contains the main building blocks that a baby needs to grow, so the main macro and micronutrients, but it doesn't contain many of these other things, like I had mentioned antibodies. Milk also contains hormones and growth factors. And this picture, again, is a little out of date. It doesn't refer to the microbiome, but we now know that milk also contains live probiotic bacteria, as well as prebiotic oligosaccharides. So that's food for those bacteria. Milk even contains stem cells, which is pretty amazing, not a focus of my own work, but something I'm always happy to talk about, as well as food antigens. And I've got that in capitals there because that's a particular interest of mine lately, because I'm interested in food allergy development. And these food antigens that occur in mother's milk can be some of the first exposures that a baby receives to allergenic foods like peanuts or fish. And we're working to understand how that works and how it might be beneficial in preventing allergies. So I really think what's important is not only to look at these building blocks individually, which is kind of how they're shown in this picture. What I think that's how research has been done in the past. You get a microbiology expert looking at the bacteria, a stem cell expert looking at the stem cells. But what I think is missing there is that we really need to look at them all together. They occur together in milk, and they're probably interacting with each other. So this new kind of philosophy on milk, the field is shifting towards thinking of milk as a system of many different biological components. And it's not only a system in and of itself, but it's a system within a system. So milk has got all these components, but it exists within this mother-infant milk triad. So the idea that the mother and infant are connected through milk, and it's actually a bidirectional connection. And so we have to think of all three of these together and how they influence the microbiome and other body systems to really understand the wisdom of human milk and how it's functioning. And so an ideal platform in which to do this would be a study of many mothers and their infants with breast milk samples. And that actually is what the child study is. So I mentioned this to you earlier. This is where I did that first paper looking at the infant microbiome. And so it was there that I got interested in breast milk and realized that we had breast milk in the freezer from all these moms whose babies we had studied the microbiome. So I was able to use this platform to study breast milk. And so very briefly, the child study is a longitudinal cohort study in Canada recruited from 4 centers, one of which is right here in Manitoba. About 3,500 families are participating. They were recruited around 2010 at the time that their mothers were pregnant. And so we've been following them, their children, their whole families ever since. This is still an ongoing study. And it was developed to answer the question of how genes and the environment influence child health and development. I mentioned allergies earlier. That's one of the outcomes addressed in the study. So really trying to understand why allergies have increased in prevalence so much in the last couple of decades. And so we are looking at genetics. We know that these conditions do tend to run in families, but at the same time, we know that's not the whole answer because our genetics as a human race haven't changed in the last couple of decades. So what has changed is the environment. We're looking very closely at the indoor home environment where babies spend so much of their formative months and years. We're looking at things that aren't as typically thought of as part of the environment, like stress or infections, social factors. We're also looking at nutrition, the outdoor environment, so things like air pollution, then of course the microbiome, which is how I got initially linked to the child study. And so the way I've been using the child study to look at breastfeeding is as follows. So firstly, asking the question, is breast milk feeding? And you'll see in a moment why I called it breast milk feeding and not just breastfeeding. Is it related to these different outcomes later in life? So allergies, asthma, but also obesity, diabetes, cognitive development. In this large cohort of mothers and infants, we're able to start looking at those associations. And then if it is, because we have those breast milk samples, we can ask the question, well, what is it in human milk that is important and driving those associations? And so there are many components, some of them I mentioned earlier. And then even beneath that, we can start to say, okay, but what is it about the moms that affects the levels of these different factors in the milk? Is it something modifiable? You know, is it her diet, smoking habits? Is it something fixed like age, ethnicity, or genetics? Or other things like probiotics, antibiotics. And we've collected information about all of these things in the child study. It's important to account for those because they might influence milk, but they might also simply influence a mother's ability or decision to breastfeed. And they might influence child health through mechanisms that are totally unrelated to breastfeeding. We can go one step further then if we identify milk components that are important and start to think about the mechanism by which they act in the baby. So is it through the gut microbiome, which was my initial doorway into human milk research, or is it a different mechanism, epigenetics, metabolism, and so on? And then my goal at the end of the day is not just to observe this in the child study cohort, but to use this information to develop interventions where we could support the next generation of mothers and babies, either to enhance breastfeeding, maybe to optimize milk composition, or to address some of those underlying factors. And then, there's lots we can do in human studies, but we can't do everything. So I do also collaborate with researchers who develop animal models to look at mechanisms and also to look at population level data. So there would be lots to say if I were to talk about all of this today. So I'll just give a few examples touching on these elements that I've highlighted in red. So the first being the connection of breast milk feeding with clinical outcomes. So in the child study, we asked a lot of questions of our families about how they're feeding their babies. And we asked it at repeated time points. A lot of longitudinal studies in the past have simply asked, did you breastfeed your baby? Yes or no? And maybe how long? But we asked a lot more questions than that. And, I won't ask you to read these, but just to give you the sense that we asked about, were they using bottles? Were they glass bottles, plastic bottles? Were they using formula? What kind of formula? And one particular question that has turned out to be really interesting is that we asked if mothers were using a pump to express their breast milk. And I wanted to point out that this actually wasn't my idea. I know that we have a lot of trainees watching today. And so Dr. Annika Klopp was a trainee, and she approached me saying, Megan, I see you're researching breast milk. I've been doing my residency in the NICU, and I see that these preterm babies are getting their mother's milk, but they get it through a tube or through a bottle. And does that make a difference? And I had honestly never thought of that. And I didn't even know that we had the data, but we looked back through the questionnaires and found this question. So we were able to look at that. And so this is one example of a study that we did together where we looked at the infants in the child study and classified them according to how they were fed. So whether they received breast milk only, all directly fed at the mother's breast, whether they received all breast milk, but some of it pumped and fed indirectly from a bottle, or whether they received formula or some combination. And you could see here that looking at asthma at 3 years of age, it was the formula-fed babies that had the highest rate, the directly breastfed babies that had the lowest rate, and the babies receiving some pump milk were somewhere in between. And to us, that was really interesting. It's not what I expected to find, but it's what the data showed. And so our next question, of course, was why? Why would that be different? And we had a lot of ideas, some of which we're still exploring, but it could be that the bioactivity of the milk is affected by the process of pumping and storage. Maybe the microbes in the milk were affected. It could be that the skin-to-skin contact between a mother and baby is important. And of course, that doesn't happen if the milk is pumped and fed from a bottle. It could be about contaminants in the bottle, the plastics. It could also be this infant-mother signaling that occurs. So these are still things we're exploring, but it's an example of the kind of hypotheses we can generate with this type of research. I do want to make my standard PSA here about pumping. Unfortunately, some of this research has been reported in the news in a way that I don't think is fair or helpful. Talking about pumped breast milk containing nasty bacteria. This really isn't the message that I think should be conveyed, but I do think it shows that pumped milk is not equivalent to nursing at the breast. However, we do see that it's beneficial compared to formula, so that's a good thing. We also know from other research that it might be the only way that a mom can provide her own milk to her baby for a variety of reasons. And so that's really important to recognize. And at the end of the day, it's important to understand why. So if we find that it's something about the way the milk is stored, we can then advise moms to store the milk differently, and so on. So moving on then to talk about the milk itself. You can see here, this is an updated version of the figure I showed earlier, because it does include those pre and probiotics. And I'm slowly chipping away at these blocks and getting them all analyzed in the child study with that goal of looking at them all together as a system eventually. So some of this work is already published. Some of it is underway or coming soon. And I'm always looking for new collaborators who measure other things. My wish list is always growing. What I'll focus on today is a little bit of data on oligosaccharides, which I have done in collaboration with Lars Bodie at UC San Diego. So human milk oligosaccharides, I mean, it's hard to pick favorites, but if I had to, I think these are one of the most important and exciting components of human milk. So if you think about milk, it's about 90% water. And of the solid components, the most abundant one is lactose, followed by lipids or fats. And then even outweighing proteins is human milk oligosaccharides. And so these are non-digestible carbohydrates, meaning they're not food for the baby. The baby doesn't digest them, but we now know they're food for the baby's microbes. And we also know that they're structurally diverse and very species specific. So for example, cows make milk oligosaccharides, but they make about 40 different kinds, whereas humans make over 100. So as soon as I heard this, I thought, well, that's amazing. Like, this is one of the reasons that human milk is so optimized to human babies. And, even though this was known, there are many different human milk oligosaccharides, there really wasn't that much known about why there are so many different ones. What do they all do? Why are they so different between different moms? And so I approached Lars Bodie as one of the world experts in studying milk oligosaccharides and said, hey, I have 2,800 breast milk samples in the freezer. We should analyze HMOs. And so we did. And the other thing that wasn't known is, okay, they're feeding the gut microbes, but what health impact does this have on the babies? And so this is something we're still actively exploring, but to show you a little bit of data. So this is Lars and my postdoc Cosetta, who worked hard on this data. Here you're seeing from about 430 moms in the child study that we analyzed the human milk oligosaccharide profiles. And you can see that all the different colors are different HMOs. From the mom who makes the least amount to the mom who makes the most, there's almost a 4-fold difference, so huge variation. And if you look at the individual HMOs, there's up to a 100-fold variation between moms. So we're still working to understand why this is. One thing we do know is that secretor status, which is a genetic trait, and you can see down here, I've separated the moms into non-secretors versus secretors. This is a single polymorphism difference, is one thing that separates moms into these kind of two clear groups, but there's still a lot of variation between them. The one thing you will notice as the main driver is that this blue oligosaccharide, 2-fucosylactose, is only made by secretors. And interestingly, this is one of the components that formula companies are starting to add to their products to make them more like human milk. And they've chosen to add 2-fucosylactose, this blue HMO that is only made by secretors. And so I think, it's an interesting question to ask. If about 25% of moms are non-secretors and do not make this HMO, is it a good thing to put it in formula and give it to every single baby? I think we don't know the answer to that. Now, is this linked to health outcomes? Well, one thing we did in the child study was to link the HMO data with food sensitization, an early form of food allergy. And what we saw was when we looked at HMOs one by one, these are little tiny graphs, but all you need to know is there was no association. We didn't find that any particular HMO was different in babies who remained healthy or who developed allergies. But when we looked at all the HMOs together using a machine learning method, we saw an important difference. And so to me, this signals that there's no single HMO that's going to predict food sensitization, but the overall profile is important. And that's just looking at one component. We still don't know the mechanism. That's something we want to learn. What happens when you add other components to this analysis, right? And think of milk as a system. And what about other conditions? So food sensitization is just one, but we could easily look at asthma and obesity and cognitive development. So there's a lot of research yet to be done. I touched on these few red points. And I mentioned that we eventually want to look at interventions. And that's something that we have to do beyond the child study, because child is an observational cohort. The kids are now 10 years old. If we want to really develop and test interventions, we have to think of new ways. And so I want to briefly tell you about two other programs in my lab. The first is MILC, the Manitoba Interdisciplinary Lactation Center, funded by Research Manitoba and the Canadian Foundation for Innovation. This is a photo from the day it was announced, and the Minister of Health and Science came to share in that celebration. And the other is IMIC, the International Milk Composition Consortium, which is funded by the Bill and Melinda Gates Foundation. And it was an exciting day for me when that was announced. This is a snapshot from the front page of the Free Press, which I know I've never been on the front page of the Free Press before. And I'm pretty sure it's safe to say breastfeeding has never been on the front page of the paper before. So I think that was a really exciting day for multiple reasons. So starting with IMIC. This study, as I mentioned, is funded by the Bill and Melinda Gates Foundation, and it speaks to that intervention piece. So the foundation had already been funding 4 randomized trials of different nutrient supplementations for mothers, and they saw some of my talks and other research and said, you know, it's pretty clear that milk is an important piece of this puzzle. So they approached me and said, if we get our studies to collect milk samples, then would you analyze them and try and understand whether the milk is changing in response to these interventions? And if so, is that important for the infant outcomes? So we connected with these ongoing studies. We're analyzing data and samples from about 1,000 mother-infant dyads. And we're looking at all the different components of milk. And then we're working with some data scientists, because when you generate these many data sets, it's not straightforward to analyze those data. So we're working with them to understand how these different milk components interact and how they influence different health outcomes. And so these data science partners are from Stanford and Berkeley, and it's been super exciting to work with them and apply these advanced methods to the analysis of milk data. And you can see that in the IMIC study, I'm getting close to the full rainbow of milk components. So it's really exciting. And the last thing to touch on is MILC, the Interdisciplinary Lactation Center here in Manitoba, which I co-direct with Dr. Nathan Nickel. Nathan comes from a public health sciences and biostatistics background and works at the Center for Health Policy. And then I have a background in human milk and epidemiology. So we're really complementary. And we're really excited about the center, which involves 3 main pillars. One is a database of information about infant feeding that Nathan is leading. And here is where we're collecting feeding information from all infants in Manitoba is the goal. We're getting there step by step. And then a human milk biorepository, which I'm leading, where we have freezers and milk analyzers and equipment to analyze human milk. And so the idea is to build up a repository of milk samples that will then be linked to the feeding data and to the population research data repository at the Center for Health Policy, so that we'll be able to understand not only how human milk is linked to maternal and infant health, but also how different population level interventions or policies are changing breastfeeding rates. So I know I'm close on time, so I'll zip through the next little bit, which is about the feeding database, which really allows many unique possibilities. We have several thousand infants already in there. Our biorepository, which is really exciting, up and running. It was born during the pandemic last April, which has been an interesting challenge this year. And then we're also, through MILC, working on some clinical trials where we're looking at, for example, the effects of different milk fortifiers on the microbiome of preterm infants in collaboration with one of the neonatologists at Health Sciences Center. We have another trial where we're looking at ways of matching human donor milk better between mothers and babies, which I'd be happy to talk more about if people have questions. And then very importantly, we have MILC Club, which is a monthly meeting of researchers, trainees, community health workers, clinicians and policymakers who are all interested in breastfeeding and human milk from different perspectives. And so we work collaboratively to do things like develop new studies, recruit research participants, and share evidence. And on that note of sharing evidence, this is the final thing I wanted to touch on, which is that I think it's really important not only to do the research, but to get it out there to the people who need it. So I do things like speak to the media. I've been a part of developing some courses for healthcare providers for them to learn about breast milk and the microbiome. I was part of a committee commissioned by the National Academies of Sciences, Engineering, and Medicine to scan the evidence on human milk and use that to develop nutrition guidelines for infants. I've developed some animated videos targeted to the public audience to get them understanding and appreciating the amazingness of human milk. And some members of my lab have been involved in writing commentaries, for example, about breastfeeding during the pandemic. Oh, I lied. I have one more exciting thing to tell you. So I focus mostly on work going on in Manitoba, but if you weren't convinced yet from that breast milk is a superfood and a personalized medicine, I have a couple quick stories for you. So this was the quote we had earlier, superfood for babies, but it's not just about babies. So there is some research going on looking at, for example, how human milk could help people recover quicker after a bone marrow transplant because it can support the immune system. So this is done in adults. There's also studies going on looking at HMOs and how they might help in treating irritable bowel syndrome, again, in adults. So we can learn a lot from human milk and apply it well beyond infants. And then, this is an example I had to give because of what we're all experiencing right now. So breast milk is a vaccine with antibodies. There's been a lot of interest in whether COVID antibodies are found in breast milk. And they are, not only in women who've recovered from COVID, but also in women once they're vaccinated. So babies, of course, are too young to get vaccinated themselves. But if their mom's been vaccinated, she's providing those protective antibodies to her baby. And so the WHO is therefore strongly recommending breastfeeding even during a pandemic. And there actually are studies ongoing to see if breast milk could be studied as a potential treatment for COVID-19. So lots of exciting research in this space, and it's changing almost on a daily basis. All right, so I hope that many of you out there are inspired to learn about human milk. If you are, there's an international society that you should think about joining. They have fabulous programs. There's a conference coming up this summer. And in particular, they have awards for trainees up to $10,000 to do a research exchange, go visit another lab, could be mine, but doesn't have to be, to learn about human milk science. So with that, I want to thank my collaborators, my team. Everything that I showed you today has been a huge collaboration, team effort. These studies take input from a lot of people. My collaborators at MILC and the Child Study in particular, and all of our funders. And I would be happy to take questions.
Meghan, thank you so much. That was fantastic and terribly exciting. I'll open the floor to questions, which can be submitted on Slido, and you can check on the YouTube link the description of how to do this, and that'll show you direct Slido URLs and the codes that you will need. But while we're waiting, I'll start off. Given my own interest in biobanking, I was wondering, Meghan, if you'd like to tell us a bit more about the MILC Biobank and its overall major goals. You talked about some of the specifics of some of the research, but if you want to talk a little bit more about how you seeing it fit into your greater research, and also another thing, maybe it's used to the greater research community, and is it designed to address the multiple components of milk, especially the bioactive components that are intriguing?
Yeah, absolutely. So I'm really excited about the biorepository. As I mentioned, the timing of it got a little inconvenient with just getting all our equipment arriving last April. So it's been a bit slow getting off the ground, but it's now fully operational. Our ultimate vision is that we'll be asking moms when they bring their babies in for their routine checkups or vaccinations to complete a quick survey, that little one page that I flashed briefly about how they're feeding their babies. And if they are breastfeeding, there'll be an additional question pop up to say, oh, and would you like to donate some milk for research? And it won't be for a specific study, it will be for this biorepository. And if they're willing, they would provide a milk sample It would come to the biorepository, and we'll also get their permission. Can we link this sample to your health information? So in Manitoba, at the Center for Health Policy, we have amazing databases that include all health information. So doctor diagnoses, prescription records, hospitalizations, and also social services data, school exams, and all sorts of things. So this is a really powerful resource for research. And if we're able to link the milk data, the milk sample, and any data we eventually generate from it to those databases, it'd be a really powerful system to look at, for example, what health conditions or medications in a mother might influence milk composition, how different milk components might be linked to infant health as that baby grows up. So this will be really unique in the world. There's nowhere else like it. There is a milk research biorepository in the US that does a lot of interesting stuff, but they just don't have that capability to link to administrative health data because that doesn't exist in the US because of their different healthcare system. So it's going to be really exciting. And then definitely we view it as a resource, not just for me or my lab or Nathan, but for the research community, so that people will be able to approach us and say, hey, I have a hypothesis or a question about milk and health or milk in a particular medication. And then they would be able to submit a proposal to access the resource. And there also would be the potential to do targeted studies. So if someone came to us and said, I'm really interested in milk in mothers with multiple sclerosis, well, then we could specifically try and recruit moms with that condition, or moms on a particular medication, or infants with a particular condition. So I view it as really quite endless possibilities. And your other part of the question was, how will we make sure that we can analyze the different components of milk, which is an important one. So we're doing a lot of work at the front end to understand the best ways to collect and store the milk so that those various different components are analyzable. And that might involve making different aliquots, processing them in different ways before they head into the freezer. So something that we're thinking about a lot up front.
Thanks. That is very exciting. I've got some questions on Slido, and the first one is, since a mother's milk is diurnal, have you looked at circadian markers that could cause these diurnal variations?
Yeah, great question. So not personally yet, but this is an area of increasing interest in the field, and it's something I'm really interested in doing. So the milk I've analyzed so far has primarily been from the child cohort study. And in that study, the milk wasn't collected at a particular standardized time of day. In fact, we asked moms to pool milk from different times of the day with the idea of getting an average, a daily average. Which is good for certain research questions, but of course it does not allow you to look at diurnal variation. So it's a question that's really interesting and a good example of something we could look at in milk if we collect the samples in the right way. It's really fascinating, like melatonin, for example, which is something that helps us sleep, is higher in milk made at night. So I think this has lots of implications too for moms who pump, for example. Should they be labeling their milk morning milk, night milk? And same thing for donor milk, where preterm babies, if their moms aren't making milk yet, they receive donor milk, which again is pooled and not labeled for time of day or other things. So really interesting area, but not something that I've looked at personally.
Okay, and another question, apart from comments like amazing talk and innovative research, keep up the good work, etc. Another question is, has there been any work done on looking at ketone bodies in breast milk and how they may correlate to any relevant outcomes?
Interesting question. So not that I am directly aware of, but that doesn't mean it hasn't been done. So I'm not sure about that one.
It is interesting because ketone bodies seem to have a mechanism associated with signaling and have some important role, it would seem, in protection against certain cardiovascular and heart diseases. I only found that out from one of our students' posters yesterday. It was enlightening and very interesting. Okay, another question is, would infants, orphans, etc., who are fed by their non-biological mothers experience the same benefits of breastfeeding?
Yeah, great question. And so this gets to that personalization of milk, right? I think probably the answer is they would still benefit. So much better to get human milk than a substitute, if possible, if mother's own milk isn't available, because there still is that humanized element of it. but it wouldn't be as personalized as if it was the baby's own mother. And so this is a question of huge interest to people in the donor milk banking field. So as I mentioned, premature babies, often their moms aren't making milk yet. And so to bridge them until the moms start making milk, we have donor milk banks. And so these are institutions that collect milk from moms who are making extra. They, at this point in time anyway, pool the milk all together and then pasteurize it because they need to be sure that pathogens aren't transferred. And so this treatment does change the bioactivity of the milk. So some proteins are probably there and still active, some are deactivated. I didn't talk today about microbes in the breast milk, but there are live bacteria in breast milk, so those would be killed, both the pathogens and the healthy bacteria. So these are all elements that if it's donor milk that's been processed, may not be as active. If it was not donor milk, but rather another mother's milk, so milk sharing is a thing that happens, then you might still preserve the activity, but it still wouldn't be paired to the mom. In that case, though, if it was like another mom directly breastfeeding a baby, you might get some of those personalized benefits because some of it's about the environment. Because that mom and baby, even though they're not biologically related, are sharing the same environment, and the mom's exposed to an infection, then the baby's going to get the protection. So there's many kind of aspects to it that I think we're still learning about. But I think it's pretty clear that human milk is going to be beneficial. And if it can't be the mother's own milk, then there are alternatives that are worth exploring.
Okay, and one last question. There are recent reports noting carryover pollutants passing from mother to infant via breastfeeding and breast milk. Is it a main concern in your study?
Yeah, another great question. So, of course, there are so many exciting and good beneficial things in milk, but there is important research happening about pollutants. So if mothers are, for example, breathing in polluted air or exposed to pesticides or chemicals, to what extent does that get into their milk and potentially get passed on to their baby? That's not something that I've personally studied, but it is an area of concern and active research. I think in our study in particular, there is the potential to look at it in child. For example, we have information about air pollution levels in different areas where the women live. We have information about the different products they're using, cleaning chemicals in their homes. My sort of personal thought on this is that cows are also breathing air and potentially pesticides. So their milk is probably not entirely pure either. And I think the many benefits of human milk are probably going to outweigh any of these potential contaminants. But that doesn't mean it shouldn't be researched and that there might not be exceptions to that for moms who are highly exposed to something particularly toxic. So it's an important question.
Okay, thank you, Meghan. I'd just like to thank you very much on behalf of all of us here for sharing all of this with us. It's exciting research, and it's wonderful to see our homegrown researcher with the incredible international teamwork and research that's going on. Certainly I'm very proud of you, and no doubt everyone else is very proud of you. Thanks again.
Thank you so much. It was a real pleasure. And I think it's kind of neat unintentionally that things have come a little bit full circle, because my first project with you, of course, was about breast cancer. And now here I am studying breast milk. So who would have known? But really appreciate the opportunity.
Okay, thanks, Meghan. And over to you, Jeff.
Thanks. Really impressive work and exciting, innovative program. So now it's my pleasure to welcome Dr. Brenda Elias from Community Health Sciences at the University of Manitoba and a founding member of the Manitoba First Nations Center for Aboriginal Health Research, to introduce our next speaker, Dr. Vikram Patel. Brenda?
Thank you so much, Jeff, and good afternoon, everyone. It is with great honor that I introduce Dr. Patel, who is the Pershing Square Professor of Global Health and Wellcome Trust Principal Research Fellow at the Harvard Medical School. He is a psychiatrist who's worked in the UK, Zimbabwe, Australia, and since 1996 in India. His work has focused on the burden of mental disorders, their association with social disadvantage, and the use of community resources for their prevention and treatment. He holds an honorary professorship at the Harvard T.H. Chan School of Public Health, the Public Health Foundation of India, and the London School of Hygiene and Tropical Medicine, where he co-founded the Center for Global Mental Health in 2008. He is a co-founder of the Sangam, an Indian NGO, which won the MacArthur Foundation's International Prize for Creative and Effective Institutions in 2008, and the World Health Organization Public Health Champion of India Award in 2016. He is a co-founder of the Movement for Global Mental Health. He has received numerous awards. He is a fellow of the UK's Academy of Medical Sciences and has served as a co-chair for the Grand Challenges in Global Mental Health on the National Mental Health Policy Group of India and the World Health High-Level Independent Commission for Non-Communicable Diseases and Mental Health. He was also listed in Time magazine's 100 Most Influential Persons of the Year in 2015. Welcome, Dr. Patel, and over to you.
Well, thank you very much, Brenda. It's a real pleasure to rejoin some of my friends from the broader Gairdner community, and I'd like to thank them for inviting me to speak with you today. My subject today is something that I have been, it's been very close to my heart for most of my career, but today my talk is going to really refer to a much broader scope of evidence around this subject, and that is to do with youth mental health. And I'm going to particularly argue that that the circumstances that affect youth mental health have greatly worsened in the last year and a half of the pandemic. I'd like to start by stating the obvious, really, that young people matter so greatly to all societies, not only, of course, because as they've been described as the world's demographic dividend, but also they are the vanguard of many social movements, from preserving our climate to combating extremist ideologies, as we've seen so vividly play out in the last few years. Therefore, their health and well-being is of critical importance, not only for themselves, but also pretty much so for the future of our planet. Now, one of the most important health concerns in young people is mental health. In fact, mental health problems together, as we'll see in a moment, account for the leading cause of the burden of suffering in this particular stage of the life course. Even before the pandemic, there were signals suggesting that there was something to worry about in terms of the mental health of adolescents and young people. This slide shows the prevalence of self-reported depression or anxiety in nearly 70,000 young Americans sampled from over 100 US colleges in the spring of 2015. And you'll see that even back then, about 10% of all young people were reporting significant mood and anxiety problems, and nearly 8 to 10% had reported a suicide attempt in the previous year. These data would not be surprising if one looked at the overall global picture of the contribution of mental substance use and self-harm behaviors to the overall burden of disease across the life course. If you look at this slide, which shows you the GBD data across the life course, on the x-axis and on the y-axis, you can see the relative contribution of mental substance use and self-harm conditions to the overall burden of disease. You look at the real peak occurs in late adolescence and the early 20s of one's life, when about a quarter of the total burden of disease is accounted for by these conditions. It is now well recognized that most mental health conditions, by some estimates, 75% of the total population morbidity of mental health problems has its origin before the age of 24. And clearly there's therefore something developmentally very, very important about this phase of life, and we'll turn to that in a moment. One of the most extreme examples of poor mental health is, of course, suicide. And in the US, suicide was already the second leading cause of death in young Americans. We often associate suicide, of course, as being commoner in older people, and that is certainly true if you look in developed countries at which age the lion's share of suicides take place. But if you look at the burden of mortality by age groups, it is very obvious that suicide ranks at the top for young people much more than for older adults. It's important to point out that the leading cause of death, unintentional injury, is also very heavily influenced by poor mental health, not least of all because of substance use. And so mental health or behavioral conditions more generally are the leading cause of death in young people. And this is true not just in the US, but also in the other country where I spend much of my time researching, which is in India, where, as you can see here, of the total number of suicides that occur across the life course, the bulk of them occur in young people between the ages of 15 and 30. In fact, suicide is the #1 cause of death in young Indians, and it has been this way for at least the better part of the past decade. What's more worrying is that even before the pandemic, the mental health of young people, if at least assessed through the suicide mortality metric, was getting a lot worse. This shows you the changes in suicide mortality rates across different age segments over the decade just before the pandemic hit our shores. And you will see that there's been an uptick across all age groups, but look at the proportionate increase in the youngest age group. Nearly 50%. And I'd ask you to pause and think about any other health condition that is associated with such a dramatic increase in mortality rates over the past decade. And I actually can't think of any one. This is not peculiarly an American phenomenon. If you look at trends in other high-income countries for which I could get data over roughly the same period, you will also see a significant increase in mortality in both young men and young women in the decade just ending in 2018 or 19. Now, many of you might wonder why, this might be the case. What is it about young people that predisposes them for such high rates of mental health problems and suicide mortality? This wonderful book, which I used as a primer to understand not just neurodevelopment in adolescence, but also to figure out how to manage my son, who was an adolescent when this book was published, wonderfully synthesizes the very complex and relatively new science of neurodevelopment that we now understand happens during the immediate years following puberty and extending into the mid-20s. When I was a medical student, I was taught that the most developmentally critical period for brains was during the first few years of life. And this is, of course, entirely true. This is, of course, the period when the brain is most plastic and most likely to respond to environmental stimuli, both positive and nurturing ones, but also harmful and stressful ones. However, we now know that there is a second, very unique developmental phase occurring during adolescence and young adulthood. And this is the phase not only when the brain is plastic, less so than in early childhood, of course, but remains very plastic, but also a stage where you see a very unique phase of brain maturation, when the prefrontal cortex of the brain, which is concerned with higher executive functions, matures about 6 to 10 years after the deeper limbic cortex, which is the part of the brain deep inside our cerebral cortex, which mediates our primary emotions like rage, anger, lust, and so on. And this effectively means that we now understand that youth are biologically and indeed evolutionarily primed to take risks and behave impulsively. Because in risk-taking and impulsive behavior is the opportunity to learn the essential life skills that one needs to learn as one transitions from being a completely dependent child, when one is completely dependent on one's adult caregivers for almost every need, to becoming a completely independent adult. Think about the fact that during the 6 to 12-year period of life, we as human beings will make the biggest social transitions that in any other comparable period of life during our life course. We will become sexually mature, become comfortable with our sexual identity, form important and intimate relationships, complete our education, find employment, and essentially become independent in our daily lives. All of this will happen in a narrow space of about 10 years. So massive social transitions. And in the way that our brains are wired and developed, we are primed to take risks and behave impulsively for precisely the reasons that I mentioned earlier. But of course, this impulsivity, which is evolutionary, also means that under certain adverse environmental circumstances, we can actually be predisposed to take risky behaviors. Those risky behaviors can have very many different phenotypes. They can range from, for example, sexual risk-taking that predisposes us to accidents, risk-taking that leads to substance use or self-harm. And you can begin to see, of course, why risk behaviors typically emerge in adolescence. The phenotype may differ under different circumstances, but the risk-taking nature of those behaviors are unique to adolescence, and we can see what the reason for that is. So what are these environmental influences that are so important? Well, one has to begin by remembering that one doesn't become an adolescent overnight. There are 10 to 15 years before this, which are profoundly important. And as I mentioned, one of the developmentally sensitive phase of life of early childhood is a critically important one to understand the origins of poor mental health in adolescence. Toxic childhood environments are now, in my mind, the smoking gun of poor mental health, demonstrated in a consistent way across populations to show a very strong association between adverse childhood experiences and poor mental health. Indeed, a dose-response relationship across a range of physical and mental health outcomes, so much so that preventing such experiences are estimated to be one of the most powerful preventative interventions for a range of both physical and mental health outcomes. Now, many of us, when we think of adverse childhood experiences, we really think of very extreme kinds of trauma, such as childhood violence or sexual abuse. And of course, those are very extreme childhood experiences. However, childhood experiences that are adverse are actually far more pervasive and common, as seen on this particular slide. Indeed, the most common adversity in childhood is growing up in a household which is struggling economically, which is finding it very hard to cover the basics like food or housing, or growing up in a household where there's parental or marital conflict. And if you look at these two leading causes of childhood adversity, you'll immediately recognize that these are far more common in every population, but especially so in low and middle income countries, not uniquely so, but especially so. And therefore, these adversities are far more common than one would have assumed if we only thought of adversity in the form of extreme trauma. The second important kind of toxic environment that predisposes to risk behaviors is in adolescence or young adulthood itself. And this slide simply shows you one dominant example of this kind of adversity. Let's remember that one of the neurodevelopmental transitions that occurs from childhood to adolescence is whereas in childhood, one is largely dependent on one's parents to shape one's immediate environment, which is why adverse childhood experiences focus on adversities that occur in the home environment. When one is an adolescent, our environment will dramatically change, both because we're now in an educational environment, but also because the key figures who shape our personalities, our mental health, are our peers. And in that context, these are extremely disturbing data, which come again from American college institutions, both private and public, nearly 100,000 in 2019. Nearly 200,000 in 2019. And what you can see here is changes in the rates of self-reported non-consensual sexual experiences by physical force between these 2 years. It's actually quite staggering when you consider, if we just look at the figures for women, here you see undergraduates and here's postgraduates. For undergraduate women in these American institutions, the rates increased so that reached 1 out of 5 undergraduate women reporting such non-consensual experiences, and that for young graduate women, it increased to 25%. These are staggering rates of unwanted sexual experiences, and these are also very toxic to one's mental health. A third kind of environment, and this is very unique, it certainly wasn't around when I was a young person, is that of the social world, social media world. Now, when I was a kid, the peer environments really were largely based on the kids who lived in the same building that I grew up in and the school in my neighborhood, which catered to kids from our neighborhood, which meant that we were roughly of the same social class and had very similar histories and stories more generally. But social media has meant that young people today have a peer network that extends far beyond one's neighborhood. In fact, it's quite possible that young people today may have more friends, digital friends, virtual friends, in the social media world than they do in their own physical neighborhood. And so it's not unreasonable to ask questions about whether this suddenly dramatically increased social network might have adverse impacts on mental health. And I'm sure we've all heard these concerns being expressed in various forms. Of course, young people are a particular risk group, but not only because their mental health is more vulnerable, but also because, of course, this is the demographic that's embraced social media the most. So what does the data actually tell us? Well, unlike other toxic environments, such as, for example, first of all, unlike other environments I mentioned before, the toxic ones of adverse childhood experiences and adverse experiences based on peer violence, social media data are actually much more mixed. It isn't the case that there is a consistent picture that emerges. These data coming from the Pew Research Center show us that actually young Americans report in roughly even proportions either a positive, a negative, or neither a positive or negative effect of social media on their mental health. And you can see a number of different examples of how social media is either experienced as being a positive influence or as a negative influence. It turns out that probably the most parsimonious explanation of the impact of social media on mental health is that what matters is not whether you use social media or not, but how you use it, what you use it for, what are the other environmental and personality characteristics of the user. So it has more to do with the individual's usage pattern and their own overall context rather than the use of social media per se. I want to also point out a number of other concerns. I won't be delving into these that young people are increasingly grappling with these days. I picked up this term eco-anxiety at a recent conference a few years ago, actually now in Stanford, where I spoke alongside a group of young people who were activists on the front line of climate change in their countries, and I discovered this term eco-anxiety, which it turns out isn't all that uncommon. I can speak, from the context of being a parent myself, of a young man who is a wildlife biologist and has been for many years extremely consumed by anxiety around the state of wildlife and conservation in the face of a range of threats, both past but also present, in particular climate change. And of course, in many parts of the world, young people are also seized by the increasing polarization that characterizes many societies. Certainly both countries that I live in, the US and India, are exemplars of such polarization. I think we all know how the killings, for example, of young African-American people, particularly young African-American men, has greatly increased the experience of poor mental health, particularly amongst African-Americans in those communities in which killings take place. And finally, the next most toxic environment is, of course, what has happened in the past year. Now, you may wonder why I've used the term thrown under the bus, and it's for very simple reasons. And that is that we have essentially, at the level of public policy, both global and national and local, essentially completely discounted the effects of containment policies, for example, the closure of educational institutions, on the mental health and well-being of children and young people. Neither have they been consulted, nor the very unique developmental needs that children and young people have to interact with their peers been taken into account. It is fair these days that now colleagues will say that it's important that in controlling the epidemic, schools should be closed last and reopened first, but that wasn't the case a year and a half ago when the pandemic first exploded. And even today, in many parts of the world, such as in India, schools have been largely shut for the past year and continue to be shut as a result, obviously, of the devastating second wave, meaning that an entire generation of children and young people have now gone without schooling for more than a year. And it's important to bear in mind that actually the vast majority of these children can neither actually access all the important social interactions that they need in schools, but for many of these young people, the main meal of their day came in schools. And of course, the majority of these young people in the developing world do not have access to their own digital devices or good enough internet for digital learning, which means their learning needs have also been thrown under the bus. We were told when the pandemic broke that the age group that was most likely to be vulnerable for poor mental health was actually older adults. Instead, the picture that is emerging from every country of the world without exception is that, in fact, it is exactly the opposite. Here you can see data from the US Census's household pulse surveys showing that there is a monotonic increase in the prevalence of self-reported significant anxiety or depression in the last 7 days by age band, so that the highest rates are in those aged 18 to 29. And as you can see, there is a systematic reduction in the self-reported prevalence the older you get, with the lowest prevalence rates being in the very oldest Americans. The differences here are more than 2 to 3-fold. Of course, it shouldn't come as a surprise that this happened, given what we know about youth mental health before the pandemic, and given what we know about the impact of the containment policies on the social lives of young people. Here is some more data on the impact of during the last year of the pandemic on self-reported distress, and you can see here again very similar results, that high levels of distress were reported amongst US adults, but particularly high amongst young Americans. A third, for example, reporting high levels of distress, and also disproportionately distributed over the population. As many of you have been reading about the disproportionate impact of the pandemic on subgroups, you will see that replicated here, with much worse mental health in young women, in young people coming from lower income backgrounds, and in young people who are single, and possibly a significant majority of them living alone. I'm particularly worried about what the future holds in many parts of the world where there has been an inadequate response at the policy level on the economic consequences of the pandemic. I think many of you will be familiar of the work of Ann Case and Angus Deaton, two economists in the US, who have been documenting the fall in life expectancy in American working age Americans, particularly white working age Americans, following the 2008 financial recession. This premature mortality was driven by suicide and substance use, mainly opioids, and they really located the cause of these increased deaths at the despair that arose out of the loss of jobs and livelihoods and ways of life consequent to the 2008 recession. I fear that unless governments around the world take very proactive actions to address the economic consequences, and indeed the changes also in the way we work and live as a consequence of the pandemic, that we may see a similar rise in many other parts of the world. So I believe this is really a historic moment for us to reframe the conversations on mental health. 50 years of research has yielded no transformative insights or action. Indeed, as I showed you earlier, if anything, the metrics were getting worse even before the pandemic, and will now perhaps escalate even more thanks to the pandemic. And therefore, we really do need to interrogate some of the models and approaches that we have used to study, research mental health, both the causation of poor mental health as well as how we address that. All of us have our theories about why we have so singularly failed in this area of health above in comparison with any other area of health. And for me, I believe there are many reasons that stand out. The first is the very narrow binary biomedical framing of mental health, in which we categorize populations into people with and without disorders into these binary boxes, which of course works very well for infectious diseases, but it works not at all for mental health, which is typically a complex a combination of traits and experiences that lie on a dimension. We don't tailor our understanding to the needs of specific disadvantaged groups of young people. We do not attend in our mental health clinical programs to the social determinants that so typically coexist with poor mental health. There's a lack of skilled providers, because we define providers as being those who have highly specialized degrees, and alongside that, a disempowerment of available resources in communities for prevention and care. And of course, young people are characterized by a feeling of invincibility and a reluctance, therefore, to seek help from professionals, especially medical professionals, and in hospital settings. So what's the opportunity? How do we reframe mental health? And what is the science that allows us to do so? There's a huge amount of science around the basic nature of mental health and its influences, which I've already touched on in the first part of my talk. But what I want to really touch on in the second part of my talk is the opportunity to respond to this burden of mental health problems in a fresh way. And I'm going to start by referring to the exercise that I co-chaired for the National Institute of Mental Health called the Grand Challenges in Global Mental Health, which was a research priority setting exercise that sought to ask more than 400 global mental health scientists about which research priorities deserve the greatest attention if we were to address the global burden of mental health conditions in a reasonably short time frame. Not surprisingly, this priority setting exercise identified implementation questions. That is to say, to do better with the science we already had rather than discovery questions. That is to say, to discover some new molecules, new genes, or new therapeutics to address mental health problems. More than $500 million, possibly closer to $1 billion of spending later, thanks to the Grand Challenges of Global Mental Health, we now know that the primary focus of action on mental health care must lie at the base of the pyramid. The base of the pyramid of care that is shown here, which is community-initiated care, community-based delivery of care through community health workers, and primary care and mental health services. And that's where much of the funding went into over the last decade of science, really interrogating questions around what kinds of interventions and how they should be delivered, deploying these non-specialist resources that are working in primary care and community settings. And thanks to more than 100 randomized controlled trials and thousands of other kinds of research studies, today I can tell you without any doubt that we know how to deliver clinical and preventive interventions for mental health problems in an effective and acceptable and affordable way in any resource setting, including where this image comes from in Zimbabwe, where the iconic friendship bench intervention has shown how elderly women in the community, whom my colleague Dixon Chibanda calls grandmothers, can be trained to deliver a brief problem-solving intervention to other members of their communities who are suffering from mood or anxiety conditions. This large body of science, by the way, on my own work, I'm not touching specifically on my work, but I've been involved with the non-governmental organization Sangat in leading more than 24 such programs of work, stretching from the prevention of mental health problems, such as for early child development and for school-based intervention for adolescents, to helping people with acute mental health problems recover, such as interventions for depression and drinking problems, to supporting people with enduring neuropsychiatric conditions like autism and schizophrenia recover from their disabilities that are associated with those conditions and really enjoy a more inclusive social world. This large body of evidence has redefined mental health interventions in many ways. What a mental health intervention comprises, typically these are now boiled down to mechanistically informed, brief psychological interventions, almost always twinned with interventions that target the social determinants that coexist with the mental health problem. These interventions can be delivered anywhere, but primarily wherever the person who needs it finds most convenient. Much of my work, for example, has been home-based, but also in other non-clinical settings such as schools. Pretty much anyone can be trained to deliver these interventions because they're very parsimonious. And so we deploy a range of different providers depending on the setting for the delivery of these interventions. And finally, the how, we focus on competency-based training, quality assurance through peer supervision, and very importantly, recognize that these community-based interventions must be embedded seamlessly with specialized care because one size does not fit all. And therefore, we need to ensure that the individual gets the care they need, starting from the community, but being scaled up or escalated to more specialized care depending on need and response. This is the evidence that's informed a number of different global initiatives that I have either led or been centrally involved with, such as the Lancet Commissions on Adolescent Health and Wellbeing and the Lancet Commission on Global Mental Health, but also the World Bank's recommendations for the scaling up of evidence-based interventions for mental health, neurological, and substance use conditions. In a nutshell, I can summarize all this body of evidence when it focuses on young people by saying that the evidence points towards effective interventions for prevention of mental health problems which target harmful environments across the early life course. I already touched on what those harmful environments were, and so how do we translate this into interventions? Well, if we look at the life course, that is the early life course, and recognize that these environments shift from very early childhood when the home environment is the main influencing factor, into for middle childhood when schools become very important, and into young adulthood when educational institutions also give way to communities, we begin to see that promoting youth mental health is everyone's business. During the earliest years of life, at home, early childhood development interventions, parenting interventions are the most evidence-based. During middle childhood and into adolescence, interventions in educational institutions, including teaching problem solving and emotional regulation skills, promoting healthy social environments in schools, and providing school-based mental health care in the form of counseling are all very strongly evidence-based. But we should also recognize broader societal interventions and community-based interventions, such as those that reduce discrimination and violence in neighborhoods, and economic interventions that support families and young people who are experiencing extreme economic difficulties. Again, harking back to the depths of despair, I believe this is the kind of societal or economic intervention that is perhaps most urgent at this particular time. Recently, the National Academy of Medicine in the US produced a consensus report on how this evidence can be translated into policy and practice for children and young people in the US. And I've just extracted one part of that report, the recommendations of the report. And as you read these recommendations, you'll begin to see many words and many ideas that are consistent with what I described earlier. For example, embracing a plurality of providers, almost all of whom are community-based, and therefore are much more accessible, particularly to young people who come from historically disadvantaged populations. Handing over leadership of these youth mental health programs to organizations in the community and developing strong coalitions. And of course, having a systematic process of monitoring the quality and outcomes of the implementation efforts. In closing then, what this evidence offers us is an opportunity to reimagine adolescent and young people's mental health in the next phase of the development of mental health systems in all our countries. First, we must look beyond narrowly defined mental illness and embrace a diversity of approaches tailored to the needs of the individual, and recognize, of course, also group differences between individuals so that we are also equity sensitive. We need to meet young people where they are, for example, either in their own homes or in educational institutions, or increasingly through the social media networks that many young people embrace. We need to focus on the base of the pyramid of care for those young people who already have developed mental health problems through the task sharing of psychosocial evidence-based, mechanistically informed psychosocial interventions to a range of non-specialized providers. We need to always have a healthy balance between individual interventions for care with structural and social interventions that really target entire groups of individuals with a primary emphasis on prevention. And finally, it's so very important that youth are the center of all decisions. This is one thing that I think has been one of the gravest concerns that I have seen in almost every country I'm aware of in the planning of public policies around the pandemic. I have rarely ever seen young people being asked about what they think needs to be done, what their role should be, and if educational institutions, as one example, must be shut down, how best can they be done to minimize the adverse impacts on young people themselves. Thanks very much. I'm looking forward to the questions.
Thank you so much. That's a terrific talk. And we're open for questions and comments. So I refer everybody to the QA opportunity, whereby just follow your YouTube direction on how to ask your question. our first question that we have is this particular participant is curious about your thoughts on the concept of anti-fragility and, sorry. Oh, unfortunately, I lost that comment.
It's on the. I can see it now.
Okay, yes, there I see it now.
So I have to say I'm not absolutely sure what that concept is. It's probably the first time I've heard it, so I apologize. I obviously am a little out of touch. If either you can just type in what you mean by anti-fragility, then I'll be able to provide a more a sensible response.
I haven't seen anything yet. So in the meantime, while they're doing that, one of the interesting aspects of your talk is the focus on youth. But what about youth transitioning into adulthood? So we do this investment in youth. We do the training at the community level and the training in community workers. But then there's this transition to adulthood. What do we do about that?
Yeah, so Brenda, first of all, can I say this is one of the most vexing issues of all, the division of the life course into these artificial categories of adolescence, adulthood, and childhood. If you look at the WHO definition of adolescence, it's from 11 to 19, but there's really no scientific foundation to that. It's a totally arbitrary age band. There's been a lot of conversations around what's a more developmentally informed age group, age range. And I think my own belief is that the more developmentally informed one is that we're talking of youth as an age range that starts from around puberty and ends in one's early 20s. And what is important about this age range is that this is the age range in which we see these dramatic neurodevelopmental changes. This is also the age range in which we see the most dramatic social transitions from childhood into autonomous adulthood. And you're absolutely right to point out that, in fact, the mental health concerns we have are far more common in late adolescence and in the early 20s than in early adolescence. And the reason for that, I believe, is because that is the period in which the greatest challenges, psychological and social, occur in terms of the transitions we make. Even up into our mid-teens, we tend to be far more sheltered, for example, because we still live with our parents, we still rely economically on them, and those are concerns that we don't directly have to deal with. But by the time one reaches one's early 20s, one has to actually pretty much assume an independent adult role in every aspect of life. And it is that transition which I believe leads to the possibility of many more stresses. But that we should not ignore the fact that the vast majority of young people make that transition very successfully. It's just that the risk factors are much greater during this period of life than at any other. And so if young people are inadequately prepared for one reason or the other, many of which I mentioned earlier, the threat to their mental health is greater during this period than at any other.
Thank you. We have one question on what is the role of genetics and all of these concerns of mental health in youth, and particularly this transition period, perhaps?
Yeah, well, there's been a lot of research on the role of genetics in mental health. First of all, it's important to emphasize that mental health problems aren't one condition. There are very heterogeneous range of conditions, from some such as autism and schizophrenia and bipolar disorder at the one end, which have a very high heritability component. And in those particular relatively rare but very severe conditions, genetic factors play a very big contribution, but yet not a lion's share. Even for schizophrenia, for example, the heritability may be 40 to 60%, which means about 40 to 60% is actually explained by environmental factors. But if you look at the far more common mental health conditions such as mood, anxiety, substance use, and trauma-related conditions, the heritability is much less, suggesting that social and environmental determinants play a much greater role in terms of etiology. That said, we should never also treat genes and environments as being separate. I think one of the most exciting areas of inquiry is how genes and environment interact with each other. The field of epigenetics is obviously the one that comes to mind here, so that genes, the expression of certain genes may be turned on or off depending on the presence or absence of certain environmental stimuli. The other important thing to remember is that whether or not genetics plays a role, biology always plays a role, because even if the condition is primarily determined by social or environmental determinants, the impact on mental health will always be through neural circuits. And so the very important observation that it is adversities in early childhood and adolescence that lead to poor mental health can now be understood through the mechanism of neurodevelopmental changes, which are actually evolutionary. They're not necessarily due to any particular genetic inheritance. This is a universal phenomenon, but that those neurodevelopmental mechanisms, plasticity, differential maturation, pruning, and so on, can be influenced by environmental factors, which therefore will shape our brain development and our mental health.
Our next question is, with unlimited access to information available to youth, do you think that anxiety associated with making the best career choice leads to poor mental health?
You know, I often wondered about that. Certainly, again, when I was a kid, we had very few choices. We also had no social media. The only information we got, other than that from our parents, was from a single channel television. So I don't know the answer to that. Seriously, I'm sure philosophers will have a lot to say whether the infinitely greater choices that young people today have, infinitely more information about so much that young people have today, whether, again, it's like social media. I don't believe whether it's a good or bad. It's going to be how you actually process that information, how much you actually balance the information you receive, so that you're able to tell false information from accurate information, which I think is a much bigger problem today. It's the filtering of information, so you know how to use information in an accurate way, which I think can be overwhelming for young people who have resource constraints, essentially. And by resources, I mean not just money resources, I also mean cognitive resources. And this again circles back to actually my belief that many of our cognitive capabilities are laid down during this first 10 to 15 years of life, provided we are exposed to environments that nurture those cognitive capabilities.
Thank you. The next question concerns about whereby you have the suicidality occurring in youth, early adulthood, but it follows some youth into their middle age and perhaps in their older years, and the continuity of care issue, and your thoughts about ways to intervene during that period of time. And one observer noted such mental health concerns that may occur in middle-aged adults, which could be a follow-up from or follow-through from their youth, or could actually happen just spontaneously, or due to a multitude of factors in their middle age.
So one of the important things that we've noticed about suicidal behavior across the life course is there seems to be two quite distinct patterns. The pattern that you see in young people is quite distinct from that you see in older and middle-aged and older adults. And it is distinct in this one particular way. A significant proportion of self-harm, suicidal behavior in young people is impulsive. It takes place in the context of acute triggers, stressful triggers. For example, the breakup of a relationship. It is often without very careful planning. This is the reason why self-harm more generally is very common in young people. And you'll see self-harm as another dimension of which suicide mortality is only the extreme end. But if you think of self-harm in its broadest ways, for example, the cutting of oneself, you will realize that self-harm behavior is actually very much more common in young people than just suicide mortality. And it is also very impulsive. It is also used by many young people as a way, in the description of young people, to release extremely distressing internal emotional experiences. In contrast, in middle-aged and older adults, suicidal behavior is much more associated with psychiatric conditions. That is to say, mental disorders like depression, substance use disorders, psychosis, and so on. The behavior, the suicide attempts tend to be far more thought through. And therefore they tend to often be much more lethal because they're actually more likely to be planned. Now you might ask why. Well, just let's circle back to my talk. And if you remember, I mentioned that the age of adolescence is the age of impulsivity, and what that effectively means is that the decisions you make when you're 16 or 17 years old are often made on the basis of raw emotions, which haven't been moderated by executive functions that lie in the prefrontal brain. Prefrontal cortex. On the other hand, when you're in your middle years, your brain is fully mature, and therefore you spend a lot more time cogitating and thinking about the actions you're about to undertake. And therefore, in paradox, suicidal behavior in middle age can actually be far more dangerous because you actually thought through this act much more with much more effort. Now, is there a connection between the two? Absolutely, because oftentimes you will find that the life course trajectories of poor mental health that you see in middle age actually have their onsets, as I mentioned earlier, in childhood and adolescence. So there is often a trajectory. That said, let me make it clear, the overwhelming majority of young people who take risks, including self-harm, have perfectly good mental health in adulthood. It is very much also a part of that developmental phase. A certain proportion will go on to experience enduring mental health problems in adulthood, but a significant majority do not. And one of the really important questions from a scientific perspective is being able to understand the predictors of that prognostic outcome. Which young people develop enduring problems, and which of them may only need support for a briefer period, and what can we do for the former group that can modify the progression of their poor mental health condition.
Thank you so much, Jeff. I just see you visually on the screen, so we're close to our very end. So I'd like to thank you so much for this tremendously important conversation about mental health, particularly your focus on suicide and youth. And I'm sure colleagues from our university will be quite excited to contact you in the future. And we have a tremendous team of researchers here at the University of Manitoba, as well as in global public health, with great interest in mental health. Over to you, Jeff.
Thank you. And our thanks to all the speakers today for a fantastic 5th anniversary symposium today. We're going to move on to the town hall portion of our meeting. And so this is really a chance to you, the audience, to ask questions of any of our guests today. They can be directed to a specific person or the group in general. They can be about the research you heard today, or any career questions you may have in terms of your experience. So we'll see what comes up on Slido here. Feel free to post there your questions. There's the link again. So maybe to start, I can just ask the panel generally if you can comment on your own mentorship experiences that really had a profound effect on your career, if you wouldn't mind sharing that with us.
Well, thanks, Jeff. Let me go first. I obviously consider the mentoring that I've received as being central to my ability to be an interdisciplinary researcher. And I think the one comment I will make about the mentorship that's been most effective for me has been mentoring from outside my discipline. I think I have learned so much from mentors who were experts in other fields, for example, in the field of infectious diseases, which might seem paradoxical that I'm a mental health researcher. But I found, and I really encourage all my mentees to always step outside of the comfort zones of their very specific discipline if they are going to be able to embrace new ideas, not necessarily new in science, but new for their field, by embracing those ideas from other fields of science and bringing it into my own. And I've certainly done that a lot by dipping into other fields of science through my mentorship.
Excellent. Any other thoughts from the panel?
Yeah, I'll just say a few words. I worked with, for my Ph.D. studies, I worked with someone who thought women didn't belong in science. And so, you can say that's a challenge for mentorship, but I learned two important things from that experience. The first is that it created this inner strength that I had, that I felt that, well, I just have to do that much better. And while that shouldn't have been the case, it really, it was really valuable for my career. It brought me to a higher level with regards to the science that I could do. And the second was that my mentor did teach me how to do a well-controlled experiment. And if there's a single graduate student who's out there, if you have to learn any one thing in graduate school, it is the best advice that I can certainly pass on, is to learn how to do a well-controlled experiment. And once you know how to do that, then you can become fearless in terms of choosing what it is that you really want to do moving forward. And so those two aspects were important, and choosing what you do want to do ultimately for a career is important. Be fearless about it and passionate.
So
Right.
I maybe share my mentorship experience. I've been fortunate to have fabulous mentors. My first clinical mentor, Ralph Feigen, most valuable pediatrician this country has seen in the last 50 years, was really the one who taught me scholarship clinically and supported me. And then Mark Fishman inspired me to be a neurologist rather than a cardiologist. I would have been a different person perhaps. But my scientific mentors are that who had the biggest influence on me, in that, just like Lainey said, doing the best control experiments, learning how to do rigorous science, think of what's an important question. I think he did me the biggest favor by telling me what not to work on. And that was really important. I went into his lab and I said, I want to work on Rett syndrome. I want to clone the Rett syndrome gene. I've seen 200 patients. I have DNA from them. I'm ready. And he goes, show me the families. And when I showed him all sporadic, he said, forget it. You can't work on that. And I said, I have to. That's why I'm in science. He said, you'll kill your career. He convinced me to pick a Mendelian disorder, the spinocerebellar ataxia. And by he convinced me to pick another, I picked spinocerebellar ataxia. But by insisting I go pick a Mendelian disorder, I was able to do something productive rather than wait 16 years for the discovery that nothing would have happened. And I was able to learn science and really sink my teeth in biology. So by him insisting in his lab, I couldn't work on my passion. He did me the biggest favor in hindsight.
So those are great stories, and I agree with everything that's been said. One of my mentors, of course, is on the line today and introduced me. That's Lee Murphy. I think maybe what I would add is the importance of networking, both to find mentors, and that being one role that a mentor can really play for you that's so important is introducing you to their network. So, the way I met Lee was actually kind of on the side of a hockey game of my brother's hockey game. So it can happen anywhere. I think it doesn't always have to be at work, which is what we often think of. But then I've had some really fantastic mentors who've helped introduce me to other people in their networks. One being Alan Ronald, who helped give me the courage to switch from cell biology into epidemiology, and helped open some doors for me in terms of introducing me to other people who then led me to other people who then led to my postdoc mentor. So I think that's really important. And I think the idea too, that you're not going to have one mentor for your whole life. You might have some lifelong mentors, but as your career twists and turns and changes, you'll need and want different mentors. So to always be on the lookout for them. And some of them you meet once a year for a conversation. Some of them you might meet a lot more often and have specific questions for. So I think Vikram commented about mentors from different disciplines. I agree, that's been really important since I do a lot of interdisciplinary research. So finding people you trust both inside and outside your organization at all different career stages. And I think mentoring can also come from, people sort of below you on the hierarchy. So I learn a lot from my trainees. Some of them come from, nursing and community health, and that's not my background at all. So I'm constantly learning from them. So I think mentorship can also go both ways.
Well, thank you everyone for sharing that. Of course, we'll have much more opportunities tomorrow in the mentoring sessions to really hear all the stories, and that's often where the most profound insights come from. So we have some questions coming back online here. I'll invite Dr. Asgar to start. She has some follow-up questions.
Well, thank you, Jeff. The question is for Dr. Huda Zoghbi. So what behavioral training paradigm do you foresee as being useful for early training of human patients to delay symptom onset?
Excellent question, because if we're going to do newborn screening and start early training during the infancy for the first 3 years of life, you have to do things that infants can do. So I can give you some examples. Girls with Rett syndrome have poor truncal tone. Motor weakness and motor planning is some of their weakness. So typically we put little babies on tummy time, just 5 minutes, and we, twice, 3 times a day, and that's enough. But maybe for a Rett child, you may have to do 5 minutes, 5 to 6 times a day. So it's really just some of the same thing you do with healthy babies, but you have to do more of it with the Rett child, because we know those neurons need that extra activity now. And same with planning, same with hand use. You may help them continuously use your hand and get more flexibility when they can use their hand. Pick, pincer grasp, drop, feed self, all of that. Typically, you let the little baby do it at their own pace. You don't sit with the little baby and focus on a task. If you want to have them retain certain words, typically we let the babies repeat here and there, okay, to do a little bit more structured time for them. So it's really the same things we do naturally with infants, let them do it at their own pace. We're going to have to do it a little bit more intensely. And because you saw with the training paradigms we did, just, 12 sessions of trainings were enough. It wasn't like massive amount of training. So just really every other week was enough, this at least in the mind. So I'm hoping that we have ways to do this.
Okay. Thank you, Dr. Zoghbi.
Thank you. Dr. Karimi, you have a couple questions?
Sure, yeah, there are a couple of questions for Dr. Fuchs. So the first one is that, let me see. Yeah. So do mature cells from skin also have inflammatory memory?
Do
mature cells in the skin also have inflammatory memory?
Yeah, very interesting. The difference between the skin differentiated cells of the skin and the differentiated cells of neural stem cells, for instance, is that the neurons that develop are long-lived. They're there for a very long period of time. And so the idea that they develop memory is very likely. With regards to the epidermis, the cells are turning over. In a human being, every 4 weeks' time, you've got a brand new body surface. And so they're just not there for a long enough period of time to be able to develop the kind of memory that we can see in the stem cells, which are there for a very extended period of time. And so we haven't looked at some of these at shorter term memory that might actually exist and be propagated during differentiation, and probably is propagated if the cells do differentiate. But it's on such a short time frame that physiologically it's probably not as important as some of these other differentiated cell types of the skin. Fibroblasts, for instance, and scarring, we really don't know how much a scarring fibroblast that might be there for a very long period of time might harbor bad memories of their experience. And I think some of those questions will be interesting as well.
Yeah, I will go with one more question, which is in the same line with this one, and also my own question, that considering stem cells actually go through self-renewal and also differentiation. So do they maintain this memory?
Yeah, we think that they do. And the stem cells of the hair follicle are actually much less proliferative than the stem cells of the epidermis. And the epidermal cells, certainly in a period of 6 months, certainly undergo divisions. And as of now, until we know more about the mechanism with regards to histone modifications and with regards to DNA methylation, we're not going to fully understand the effects of cell division on a memory-induced phenomenon, and I think it's an area that is intensely of interest with regards to understanding memory for the purpose of being able to ultimately erase memory. Once we understand enough about it, ideally we can erase it in the bad conditions and keep it in the good conditions.
Yeah, interesting concept. For the interest of time, so we will stop to provide opportunity for other speakers. Thank you.
Thank you. So Helen, we have a question from Slido to anyone on the panel. Do you have advice on how to approach learning about new topics, having little prior knowledge yourself, and how to gain new perspectives from different disciplines? Open to anyone.
I'm happy to start because that has been the story of my life.
Okay.
So, when I started, what I learned in arts lab is molecular biology and how to map genes, human disease genes. But when I found the ataxia gene, the next challenge was to now make a mouse, do biochemistry, totally new field for me. So the best way I found that worked, you go to the best person in your institution who does that. I went to Alan Bradley's office and I said, I want to make him a knockout mouse. It was early days for knockout technology. And I want to watch you do it and I want to learn how you do it. And you'll be collaborator and we'll do it together, and then I will learn how to do it. And that's really how it happened. I walked to Steve Elledge's lab and I said, I want to understand the biochemistry of ataxin 1. I know nothing about biochemistry. Let's sit down and talk. How do you find protein interactors? And we would talk and so on. So it's really that's what works the best. And then you give back. Once I knew how to do knockouts, I made knockouts to so many other younger colleagues in the institution. And it comes back to you. Then I needed to evolve into fly work. Then I need to evolve into neurophysiology, and my colleague will help. So it's really give and take. When you collaborate with someone, tell them you're gonna acknowledge their contribution, learn from them, give back, then the next thing you wanna learn, there'll be somebody to teach you the next week. At least that's how I've done it. Very practical.
Maybe I'll build on that too, because Huda and I have very similar experiences, except that as an undergraduate, I was, my major was in physical chemistry. I almost went to graduate school in that area. And so for me, going into molecular biology and biochemistry was a complete eye-opener. And that continued. having that training, I think what, again, I'll reflect on is that importance of really learning how to do experiments. And once you know that, then you can really, I think, apply it to many different fields. I completely agree with Huda that when we started to work on cloning, that was new for me. And we went to contact people at UCSF and Genentech, which was just developing, and said, we need protocols. When it came to transgenic mice, we didn't have anybody on our campus, but there was someone in another campus in Chicago, and we just set up shop in her laboratory and learned the technology, and then brought it back to the university. So I think it really is. It's part of the excitement about science, is that you're constantly choosing the questions that you're interested in and passionate about knowing the answer to, and wherever it takes you, those are the directions you should follow. And there's always a way to learn from, to learn how to do it.
Yeah, I would agree. I feel like Huda said, this is the story of my life. So I started out in biochemistry and cancer, and I've ended up in microbiome and breast milk. Like, they are very far apart. But getting here has been so interesting. And just like you said, Elaine, I think that's what's exciting about being a scientist, is that is what our life is finding out new things, learning new things. And I do think collaboration is a big part of that. So, I think I told the story of hearing about HMOs and thinking, I have breast milk, I need to study HMOs, but I don't know how to do that. So, I was at a conference, I walked up to the speaker afterwards and said, hey, like, we should work together. And we, wrote a grant and were able to do that. So finding people who are experts and who are good to work with, I think that's also a litmus test to do, like, make sure that not only this person is good at what they do, but that you get along and would enjoy working with them. I think also something I've learned over time is when you start out into a new thing, there's a tendency, at least for myself, to have this imposter syndrome and feel like, I don't know what I'm doing here, and therefore I must not have anything useful to say. But I found that actually that's where you can get some of the best ideas, is when you're brand new to something, because you have a totally fresh perspective. And I see that now with people joining my team, where they've never studied milk before, and they come in and ask some crazy questions that are like, oh, why did we never think of that? But it's because they're a bioengineer, or they are, somebody totally different. So looking for people to help you learn those new things, and then having the confidence in yourself to, the motto in my lab is like, there's no such thing as a dumb question, because I think that's where the exciting discoveries and ideas happen.
Great, thank you. Dr. Patel, would you like to join in?
Well, no, I agree with everyone, but I wanted to add that there is one of the best things that's happened in the last year has been the ability for people to dip into international meetings or conferences without having to travel anywhere. And in many ways, I think this opens the door not just to stepping into someone's lab, but actually stepping into someone's world by simply, registering. Many of these things are actually free now or available free after the conference is over. So to actually get a flavor of science from other disciplines, one doesn't have to invest all that much anymore. And I myself have found myself logging into lectures online on topics that I want to learn about. And I found it incredible to actually hear a master in that particular field, present from the convenience of my own home. And so I think that's something else. I hope that's one of the things that's interesting, that we may not fully give up when we go back to in-person. I think the way this has improved access and equity for people to participate, particularly younger researchers, I think it's been quite transformative.
That's a really good point. Thank you. Lee Murphy, you have a follow-up question?
Yes, these are for Meghan that were on Slido. We couldn't get around to. You mentioned hormones, and is there any relationship between breastfeeding and puberty?
So I'm inferring that the person means being breastfed as an infant, and then onset of puberty when that breastfed child grows up. So Yes, hasn't been an area of my research, but I think there is data on that, and in particular from other animals. So one interesting thing about breastfeeding research that I've learned is that there hasn't been, kind of surprisingly, all that much research on human milk and human breastfeeding. But there has been a lot on cow milk and other livestock, because that's a huge industry, right? The dairy industry. So to our last point about connecting with people in other fields, it's interesting because those two fields don't often talk to each other, and yet there are many lessons to be learned. But I'm thinking about research in piglets where they've shown that exposure to lactation and milk, mother's milk, as a piglet has a really big impact on the onset of, I don't know if it's called puberty in a pig, but when that pig becomes fertile and how many piglets they will have in each of their litters. So this kind of transgenerational importance of metabolic programming through breast milk. We've done in child some analysis of insulin, adiponectin, and leptin in milk. So these are hormones that are related to satiety, metabolism, weight gain. They've been studied a lot in serum in adults for questions related to metabolism, and we're starting to learn about their role in breast milk. And it's really interesting to think about because insulin, as one example, I mean, scientists have tried for decades to generate oral insulin for diabetes patients, and it's never worked. As you know, people still have to inject their insulin. And yet babies are consuming insulin orally in milk. And insulin in milk is like 100-fold more concentrated in milk than in serum. So you have to think that the mother wouldn't invest the energy in concentrating insulin in her milk if it didn't have a role in the infant. And so we're learning that infants express insulin receptors in their gut, which is not the case in adults. And also they have a leaky gut, so that insulin can be absorbed into circulation in babies, whereas it can't be in adults. So there's all sorts of fascinating things about hormones in milk, and we're still learning.
Okay. And another question is, comment was very exciting talk. And have there been any risks associated with feeding babies with human milk stored in the bank for a significant length of time?
Interesting question. So not my primary of expertise, so I couldn't say for sure, but not that I'm aware of. However, there is a sort of not super evidence-based recommendation that donor milk is not accepted from moms over a certain, not age, but a certain time, a certain baby age. So I think, and it might vary by milk bank, but some milk banks, for example, don't accept milk after the donor mother's baby is a year old, because there's concerns about whether that milk is still ideal for a preterm infant, since it does change over time. But it's a little bit more of a, well, we think that that's probably the right thing to do, but there's a lot of research going on now to really understand what is the composition of milk later in lactation. Is it still okay for preterm babies? So that's still something that is being studied in terms of being in the bank for how long it's been in the freezer. Milk banks do have sort of standard protocol that after a certain number of months, the milk wouldn't be used anymore. They try to get through all the milk before it gets to that stage, of course, so none goes to waste. But the banks do have protocols for that.
And there's one other question, and I think it means they're asking, does the infant before birth influence the mother's milk?
Yeah, that's really interesting and something that we're still learning about. But for example, milk made for a son versus a daughter is different in some components. So the fetal sex seems to influence the production of milk, and whether that's something from prenatal programming and impact on the mammary gland or something more postnatal is still unknown. But I think that signals that, yes, some things happening before delivery could actually be influencing milk composition. But again, still something we're learning about.
Okay, and I think that's all that were left over from your talk.
Thank you, Lee. Well, we're at the top of the hour, so I'm afraid my sad duty to close the ceremonies for this year. Our thanks, generous thanks to all of the speakers today for taking the time and sharing your research and your thoughts and research with the graduate students of Canada and CSHR. If we could all clap, you'd hear a roar of that right now. I'd like to also give thanks to Dan and Chris from IST for making this Zoom version of the Gairdner Symposium such a success today. This is working on the fly and being very good at your craft, and it appears seamlessly to us, so thank you. Of course, thanks to our major sponsors, CSHR, the faculties of medicine across Canada. And a special thanks to Jessica Fraser, who organized much of this symposium on the ground. And of course to Ed Krieger for his unfailing dedication to this symposium for 34 years now. Fantastic year after year. Thank you, Ed. And lastly, thanks to Janet Rossent and the Gairdner Foundation for your support. This has been a wonderful partnership for 5 years now. It's really elevated what was already a wonderful national symposium. So thank you again for your support. Janet, I don't know if you want to have a closing comment.
I guess I could just say that the final few words. I mean, I have been listening in and to all the talks. It's fantastic. It's wonderful to hear our Gairdner laureates. It's wonderful to hear Meghan from our adjudication committee. So we're looking at all aspects of how Gairdner people get involved in science. And it's incredibly exciting. And I think the mentorship we just heard about is going to be very important as we go into the mentorship sessions tomorrow. A lot to be learned from everybody. So thanks to everybody, and it's been a great time.
Thank you. So that concludes today, but the symposium still goes on for tomorrow and Friday. Enjoy the rest of the week, and we'll see you next year.